Good morning, good afternoon, and good evening. It is my pleasure to welcome all the participants from all over the world for today's International Virtual Symposium. I, Dr. Rajiv, on behalf of Vokkaligara Sangha Dental College and Hospital, Bangalore Academy of Periodontology, and Rajiv Gandhi University of Health Sciences, welcome all the eminent speakers for the today's symposium. Professor Kalsha, Professor Ramesh, Mr. William Johnston, a research scholar from University of Glasgow, and Dr. Mira, a senior scientist at the Center for Advanced Research in Public Health, Spain. I welcome our principal, Dr. Rajkumar DC, who is a renowned oral and maxillofacial surgeon for symposium. I call upon Dr. Rajkumar DC to say a few words.
Thank you, Dr. Rajiv. I welcome all the distinguished speakers and participants for today's program. It's an honor to host the International Virtual Symposium on Oral Microbiome in our college. Our college was established in the year 1986 and is considered one among the top dental institutions in the country. We offer Bachelor of Dental Surgery, Master of Dental Surgery, postgraduate diploma courses, and PhD programs in various specialties of dentistry. It's a matter of pride that our alumni are scattered globally, providing quality dental service. I take this opportunity to congratulate the Department of Pedodontia, VS Dental College and Hospital, for organizing this symposium, and I wish all the success for today's symposium. Thank you.
We are proud to announce that the symposium is being conducted in association with Rajiv Gandhi University of Health Sciences and Bangalore Academy of Periodontology. Both the organizations are in their Silver Jubilee year. Bangalore Academy of Periodontology, in association with Rajiv Gandhi University of Health Sciences, are conducting various CDE programs as part of Silver Jubilee celebration. I request all the participants to make full use of these programs and contribute to their success. We all know the role of oral microorganisms in the maintenance of oral health as well as systemic health. Understanding the role of the oral microbes will help us in rendering personalized and precision treatment. This international symposium with all the distinguished speakers will throw light on the various aspects of oral microbiome. Before starting the program, we will invoke the blessings of the Almighty by lighting the lamp.
Shriyansh, can we? Kalyanam, Arogyam Dhanasampada, Shubham Kurutvam Kalyanam, Arogyam Dhanasampada, Shatrubuddhi Vinashaya, Deepajyotir Namostute, Deepajyotir Namostute, Shubham Kurutvam Kalyanam, Arogyam Dhanasampada, Shubham Kurutvam Kalyanam, Arogyam Dhanasampada. Thank you, Shreyansh.
Thank you, Shreyansh. Today's lectures will be moderated by Dr. Sushma R. Galgali, Professor and Ed, and Dr. Aruna D.R., Professor from Department of Periodontics, VS Dental College and Hospital, Bangalore. Both the distinguished moderators have vast experience in the field of periodontology. I will call upon Dr. Sushma R. Galgali to moderate the first two lectures. Thank you.
Thank you, Dr. Rajiv. Greetings to you all from VS Dental College in Bangalore. It gives me immense pleasure to introduce the first speaker of the symposium, Dr. Mira. Dr. Mira is a senior scientist at the Center for Advanced Research in Public Health, Spain, where he leads the Oral Microbiome Laboratory. He studied biology at the University of Alicante, where he got the Degree Excellence Award and specialized in evolution at Oxford University as a British Council Scholar, where he obtained an MSc degree. He then stayed at Oxford for PhD as a Rhodes Scholar, working on bacterial symbionts and evolution of mitochondria. Later on, he continued his research career at the University of Arizona, funded by the Fulbright program, doing research on bacterial symbionts and bacterial comparative genomics with Nancy Moran and Howard Ockman. He then moved to Sweden as a postdoctoral researcher in Steve Anderson's lab at Uppsala University, where he worked on bacterial genomics and microarray studies in intracellular parasites. He then returned to Spain as a Ramon y Cajal researcher, working in the evolutionary genomics group at Miguel Hernandez University between 2003 to 2008. After this, he got a permanent position of a research scientist at Center for Advanced Research in Public Health, where he is currently based. He is one of the founders of the Spanish Network of Bacterial Genomics and the coordinator and a member of the directive board of the National Association of Ramon y Cajal Scientists. His main areas of interest are study of human microbiome, particularly the oral cavity, evolutionary and comparative genomics of bacteria, and together with Joseba Vicandi, he is developing online genomic tools to study oligonucleotide frequencies and other DNA composition features at the genomic scale. Today, he will be speaking on development and composition of human microbiome, dysbiosis, and its implication on oral diseases. Over to you, Dr. Meera.
Thank you so much. Can you hear me?
Sure, we can. You're audible.
Okay, I'm going to share my screen. Okay, can you see my screen and my mouse?
Yes, we are able to view your screen.
Okay, thank you very much for the invitation to be here. It's a very big pleasure to be here today to talk to you about the research that we have been doing in the last years in my lab. And it is, of course, a very big honor to be opening the talks today. I think, you know, we are working on the oral diseases in general are some of the most widespread diseases in the world. You know, it is estimated that between 80 and 90% of the population around the world, for example, have got in the past dental caries, and the levels of gum diseases are also extremely high. And however, even though they are caused by organisms, by bacteria, we still, in the era of vaccines, antibiotics, and so on, we have still not been able to fight these diseases. So why is that? You know, why are still oral diseases around us? One of the reasons is that these diseases are extremely complex. So we don't have a single causing agent, as it happens for most infectious cases. But in oral diseases, actually, there are many of these organisms. So, for example, in the case of gum disease, we have at least the so-called red complex, at least 3 species which are involved, these 3 ones here. And in the case of dental caries, almost 100 years ago, Streptococcus mutans was isolated from the cavity of a patient, and it was found to be able to produce acid. It's also acidophilic. It enjoys being an acid. It was able to attach to the surfaces of the teeth, and it was considered to be the causing agent of caries. But then there were many other species, some of them actually found in the last 20 years or so, including Escardovia, which actually happens to be one of the main causing agents in early childhood caries. So we don't have a single species, but, you know, a group of different species that can be involved. And this, of course, makes it very difficult to study and to combat. The second big reason for this is that the oral ecosystem is extremely complex. We have over 500 oral species, some of them attached to the tooth, others to the gums, to the tongue. And then, of course, when they attach to the surfaces, they are able to form oral biofilms. And these actually change a lot from the beginning to, you know, after you brush your teeth, for example, some of the bacteria are going to attach to the surface. And then if you take a sample several hours later or 24 hours later, the complexity of the biofilm is going to be very, very difficult. It's going to be very different, and therefore even the time of sampling is going to be very important to have a good sample to study the diseases. And of course, most of these organisms actually appear to be good for us. So only some of them are going to be causing the diseases, and it's going to be therefore very difficult to keep the balance, trying to direct our efforts against those oral pathogens, but keeping the rest untouched. And then to make things even more difficult, it has been estimated that at least half of the species we have in our mouth have not been cultured yet. And as you know, the basis for the field actually has been to be able to culture individual species in an agar plate and being able to study. So actually we are not able to do that. And that's why the last 20 years have been extremely exciting in the field because of the DNA studies. So we have been able to do metagenomic studies where we are actually able to study the DNA of all the organisms present in a sample, obviating the need to culture them. And this has been very important because it has given us really fascinating insights about how oral diseases are caused. A big part of the studies, actually what we do first when we have an oral sample is to extract the DNA and RNA from the sample, and then by PCR we can amplify a particular gene which is present in all bacteria, for example the 16S rRNA gene. And then now with the new methods for high throughput sequencing, we can actually do massive DNA sequencing of that amplified PCR product. We compare those sequences to the databases, and then we can actually estimate which organisms are there. It has been very important because suddenly from only being able to culture a few species, now we can actually have access to most of the bacteria present in the sample. And apart from this high throughput 16S sequencing, we have some other approaches that actually have been very important in the last years to get insights about the oral diseases. So we can actually get a sample, extract the DNA, and not only know who is there by sequencing the PCR product, but also we can actually know what is all the DNA present in a particular sample. This is what we call the metagenome. So we actually sequence the total DNA, and we know not only who is there, but what genes they have, what is their genetic potential. And if we want to have a more functional approach, we can extract the RNA, which is the active part of the genetic material, and then we can sequence the total RNA content in a sample, for example, dental plaque or a cavity or a pocket. And that's what we call the metatranscriptome. So we can have access to all the genes which are being expressed at a particular moment. And the same if we do the metaproteome or the metabolome, where we study all the proteins or the metabolites which are produced by that community as the outcome of their metabolic pathways. So this means that we have to do lots of analysis, and actually the main bottleneck now that the sequencing, for example, has become very inexpensive, it has been actually possible to do this kind of studies, and the main problem actually lies in how to analyze the data. So this is an image that is not ours. I took it from the internet because it looked more fancy, and this that doesn't look so nice, is actually the cluster that we have at my institute. It's a computer cluster with 250 nodes, and that means that we have to compare the DNA sequences that we obtain to the databases. If we do that in a normal computer, it would take months. And here what we do is to, we are able to split that computational work in parallel in between the different nodes, and therefore we can do that in just a few hours. So this is very important, but it means that the kind of life that I thought I was going to have, like this one, like Louis Pasteur and the other people working in the field more than 100 years ago, looking at the organisms under how to culture them in agar plates and so on. Actually, we do that, but it's only just a part of our work. But most of our time we spend it like this. So I know it doesn't have the glamorous life, that I expected. But actually, I'm going to try to show you today that this is not as boring as it looks in this picture, and we are actually at a really incredible moment in time in history where we are able for the first time to look at all the bacteria present in a particular sample, to know which genes they are expressing, how do they change when they infect, how they can evade the immune system, and so on. We are really at a extremely exciting moment, and I'm going to try to summarize some of the results that we have been able to get using these new techniques. I think this image illustrates very well this change that we have suffered, that we have undergone from culture to the DNA sequence era. This is a work that we did a few years ago, where Inma Thomas took blood samples from patients after the wisdom tooth was extracted. And after doing that, you know that some of the oral bacteria actually can enter the bloodstream, and that can cause problems. How do we know if we have oral organisms in blood? Well, you know, it's even today in hospitals, the way that this is done is just by culture. So what we did is that with half of the sample, we culture that blood sample and the different culture media with and without oxygen and so on. And what we have here on the top of this iceberg is what we found. These are the 4 genera that we were able to isolate. The most common one was Streptococcus. So here the size of the word is related to the proportion with which we found those organisms in the sample, in this case in the culture. And now with the other half of the blood, we extracted DNA and we sequenced it. And this is all the bacteria that we actually found. Look at the incredible array of organisms that we found that we could not see by culture. Why is this? Because in the culture, our culture conditions, even if we try to have very different culture media and so on, we are going to favor some organisms which are able to grow very well under those conditions. However, here we have the total the diversity that is present there. And not only there are many organisms that we could not see, but actually we were extremely biased in our idea of what organisms were present in blood, for example. So we thought that Streptococcus was the most common one, but when we look at the DNA, it's only a small portion, as you see here, where I'm marking with the arrow. So it's only a small portion, and we were really biased. We were not only seeing just the tip of the iceberg of diversity in our samples, but actually thinking that the most important organisms in terms of their levels were actually totally biased. So now I think if we apply these kind of techniques, not only to these blood samples, but within the cavities, in the tongue, in the gums, in the pockets, in the people with disease, I think we now have the tools to be able to really understand much better what are the causing agents of the disease, whether we have organisms which are able to protect us, and so on. But not only the DNA high-throughput sequencing and omics techniques have been important to unravel what happens in the oral cavity. There are also some very important advances like this one, which is a sample of dental plaque taken between 12 and 24 hours after toothbrushing. And we have here, we are able to see here the structure of the biofilm by a technique called classic fish. In the past, we could use this fish technique with which we could label between 2 and 3 different organisms, each of them with a different marker. But actually, now we can do that with 10, 20 different organisms at once. So here we have actually 10 oral bacteria which have been labeled with a different color, each of them. And it is amazing because when we saw this, the part on the left here is actually the part of the biofilm which is attached to the teeth, and the part on the right here is actually exposed to the oral cavity. And the first thing that really, is really amazing is that most of the architecture of the biofilm is formed by this species that we know very little about, and it forms these very long filaments going from the surface of the teeth to the outside, forming this so-called hedgehog kind of structure. And then at the edges of the filaments, we have here marked in green streptococci, which attach to the edges, to the end of the filaments. And of course, these organisms are able to endure oxygen, so they form somehow some kind of a shield around the oral biofilm that protects the inside of the biofilm, being able to create an anaerobic atmosphere here in the middle, where some strict anaerobes are able to grow, like here in white, for example, the Fusobacterium and others, which are strict anaerobes and are able to survive within this organization, So I think that it's the architecture of the biofilm is more complex than we thought, and is more organized. And when I look at this, it actually seems to me like an Amazonian rainforest, but at the microscopic scale. So this is probably not only true for oral biofilms, but also for other niches in the human body, for example, in the gut, probably. But this is the first time that we have been able to see in an oral sample. This is, of course, what we had in mind before this about how the dental plaque was formed. We have some organisms that appear to attach early to the tooth surface, and then we have a set of interactions between them that allow specific species, to attach to each other until we have here this very long organism, Fusobacterium, which appears to be a bridge between the early and the late colonizers of dental plaque. We have an image here of one of these specific co-aggregation patterns between Fusobacterium and P. gingivalis, which is one of the main causing agents of gum diseases. And of course, this is very interesting because it appears that these very long organisms is like a place where many oral pathogens are going to attach. So in a way, we don't need to target, for example, P. gingivalis. In theory, should be enough. If we are able to target this organism, we avoid it to attach to the biofilm, and therefore we would prevent a fully formed biofilm with oral pathogens to be formed. This is what we have been trying to do, and Arantxa Lopez, one of the postdocs that was in my lab, actually found an antibacterial peptide which is able to specifically kill this organism, Fusobacterium, and therefore the rest of the oral pathogens are not able to attach. And we have shown that it actually works in vivo. So I think this is why actually knowing very well the basics of the oral microbiology is going to help us to design new strategies to combat the oral biofilms. This is a scheme that I like very much because it simplifies a lot how oral diseases actually arrive. In the left here, we have this circle of dental caries, and I just want to emphasize that there is always like a disease driver which is going to start to trigger the oral disease. In the case of dental caries, it's going to be the consumption of sugars, and what happens is that some oral organisms are able to ferment those sugars, and therefore when they produce organic acids, this is going to make the pH to get lower. When the pH gets more acidic, that is going to select for acid-tolerant organisms, and they are going to produce more acid because they are the ones able to ferment the sugars, and therefore we have a vicious circle here, a positive feedback loop, and that's going to make the disease to start, and they could end up having cavities. In the case of gum diseases, the initial factor that might trigger the disease could be, for example, a lack of proper oral hygiene. That's going to make that when the plaque accumulates, then we are going to have gingival inflammation, and the body is going to react by having this protein-rich gingival serum that is going to select for organisms able to feed on the proteins, and organisms that actually like the inflammation. So these inflammophilic organisms are going to increase in proportion. They're going to accumulate more plaque and to create more inflammation. And again, a vicious circle is formed, giving rise to gum diseases. So there is always some external factor, and we have to control those factors, because the main point that I want to emphasize today is that we have to try to keep the balance. Of course, it's very important to study oral health, not only because of oral diseases, but also because they have an effect in the whole body. We have here some of the general effects that have been found in the past, including an influence on blood pressure, and even, for example, an influence on the rate of preterm birth. But apart from this, in the last years, the research has been really important because it has put oral health in the center of of general body health. So, for example, there has been very important experiments where it has been found that Fusobacterium, this organism that inhabits the oral cavity, if it actually is able to reach the gut, it's able to induce colon cancer. This is probably the second case in history where an organism has been found to be able to trigger cancer. The first one was Helicobacter, which, as you know, is able to cause gastric ulcer, but also gastric cancer. And then there is more and more evidence that several other yeast and oral bacteria, they can make intensive chronic inflammation that may actually end up initiating cancer. So there are even in vitro studies where healthy cells are being able to be transformed into tumor cells due to the presence of this organism. And a couple of years ago, what they did actually in a mouse model of colon cancer, they treated the mice with antibiotics. They were able to get rid of these organisms in the tumors, and they were able to stop the development of the tumors, and even being able to eliminate over 50% of the tumors that these mice had. So this really opens the window to understand, and we have to be able to transmit our patients actually that having a good oral health is very important for your general systemic health. Another case was published a couple of years ago because the gingipines, which is an toxin which is going to be released in the middle by P. gingivalis, was found not only in the gingiva, but it was actually found in post-mortem brains of people that died having Alzheimer's. And actually, we have here a section of these post-mortem brains with a stain of the gingipines, and they actually appear to be very common in the brain. When we look at this under a larger magnification, we see the normal structure of these P. gingivalis cells. And actually, they appear to be more or less in the same places where the amyloid plaque places are. So this opens the chance that actually some oral bacteria can go into the brain and could be involved in these diseases. And this is just started, but there's a whole new field. And I think we have to emphasize how important oral health is for the general health of the body. But of course, there are not only all these organisms causing the disease in the general health, but also we have some oral bacteria which are very important for the general health. And we have here the example of some oral bacteria that are able to transform nitrate into nitrite, so NO3 into NO2, and then nitrite into nitric oxide. This is the work that has been doing my PhD student, Bob Rossier, and has been really incredible because the nitrate is present in the vegetables that we actually have in our diet, also a little bit in fruits. And what happens is that this ability to transform nitrate into nitrite, the human body doesn't have the enzymes able to do that. So we rely entirely on the oral microbes to be able to produce nitrite. Then the nitrite can be transformed into nitric oxide, which has very important functions in the body. For example, nitric oxide is a very important vasodilator, and therefore, if we don't have oral microbes able to perform this function, our blood pressure is going to increase. And of course, one can think, well, how long are the oral microbes going to be in contact with the nitrate from the diet to be able to perform this function? And of course, it's just going to be for a few minutes while we actually chew our food. But then, and this is when it becomes really interesting, when we swallow the nitrate, part of it is going to be lost, but a big part of it is going to be recycled by our body, and the nitrate goes into the bloodstream. And then when it passes by the oral cavity, the salivary glands are going to capture this nitrate and to concentrate it into the saliva, which means that 1 hour after having a vegetable-rich meal, the levels of nitrate in saliva are going to increase a lot, and they are about 10 times higher than the levels of nitrate in blood. So our body through human evolution, has made sure that actually is able to provide nitrate to the oral bacteria, so we can have good levels of nitric oxide in our body. So this is the way it actually works. Imagine that we swallow, for example, a glass with beetroot juice. Beetroot has very high levels of nitrate. Then when we swallow it, it's going to go into the bloodstream. It's going to be captured by the salivary glands. And then we have high levels of nitrate in saliva for a few hours. There, our oral microbes, some of them, are able to transform nitrate into nitrite, and then the nitrate into nitric oxide, either by the bacteria or by our own body. The pass from nitrite into nitric oxide is also able to be performed by the human enzymes, and then the nitric oxides into the bloodstream is going to release, is going to have an effect to lower our blood pressure, among many other positive functions in the body. So this is what happens when we swallow beetroot juice, for example, with high levels of nitrate. We have here an individual that after half an hour has very high levels of nitrate in saliva, and then is going to be kept there for a few hours. We have here another individual which actually is not very good at recycling the nitrate. So in this particular individual, most of it is going to be lost in the urine, but it still has some levels of nitrate. And here we have two individuals that vary in the levels of these oral bacteria, which are able to reduce the nitrate. So I have one here with very high levels, and you can see the levels of nitrite here, NO2. Half an hour after we ingest the beetroot juice, we have high levels of nitrate, and then 1 hour after, we have high levels of nitrite, NO2. And here we have another individual that has gum disease, and we have seen that actually individuals with gum bleeding and gum disease have a really spectacular drop in the levels of these good oral bacteria. And as a consequence of that, people with gum diseases actually are not able to produce nitrite. They are going to have a chronic deficit in nitric oxide, and therefore we believe that that's going to make them to have an increased risk of blood pressure and cardiovascular risk. Okay, how are we going to acquire these oral organisms? We know that we are going to acquire them from the mother, from the environment, from the food, and so on. And we wanted to explore this in more in detail. And we were very lucky to have access to this set of oral samples that were that people in Sweden, had been taking for years. They actually followed a cohort of about 100 children for 7 years. They collected fecal and oral samples after birth, 1 month, 3 months, 6 months, 1 year, 2 years, 5 years, and 7 years after birth. And then those samples were frozen for years in the freezer, and they asked us to analyze them, and has given us an extraordinary chance to actually understand how this process of acquiring the new oral microbes actually happen. Actually, they had a very excellent clinical record, and we actually knew what they had eaten, whether they were born by C-section or by vaginal birth, whether they took antibiotics, what kind of food, how long did they breastfeed their children, and so on. So integrating all this information, and this was the PhD thesis of Maida Siddiq that finished a couple of years ago in my lab, we were able to understand how these organisms actually are able to colonize the oral cavity. And we have here these graphs at 3 months, 6 months, 1 year, 2 years, and 7 years. And we see that some bacteria are present from the very early months. So we have at the beginning very high levels of streptococci and lactobacilli, which actually are present there only during the time of the breastfeeding. And of course, these organisms are going to be able to transform the lactose from milk into lactic acid. And then probably due to that, we have, after a few months, an increase in this organism, which actually feeds on lactic acid. So what we see here is that organisms that use organic acids as a carbon source are going to appear after we have other organisms able to produce those organic acids. So therefore, there is a succession of organisms, and the action of some oral organisms are going to change the environment, allowing other bacteria to be able to settle down and to colonize. There have some bacteria which appear to be from the beginning, but they increase steadily through the years. And we have some organisms that only appear after 1 or 2 years with the when the first teeth arise, and also with the introduction of solid foods and so on. So we have all this succession of organisms, and we could actually study which clinical factors and environmental factors are going to have an influence on the oral organisms that are able to colonize. And we see that one of them is whether the baby was born by C-section or by vaginal birth. And this is important during the first year of life. So we have some bacteria like actinomyces, which actually appear to be at significantly lower levels in children born by C-section. But after 1 year, what we see is that they actually, again, are able to converge, and then we don't see any significant changes between people that were born by C-section or by vaginal birth. There are other factors, actually, that do have a long-term influence. And the most important of all of them, we found it was the length of the breastfeeding period. So these were all Swedish mothers. They all breastfed their babies at least for a month, but some of them breastfed them for 1, 2, or 3 months, and others for 6 months, even for a year. So when we actually look at the oral bacterial composition in children that were breastfed only for 3 months, or children that were breastfed for a longer period, up to for 12 months, we see that during the first 6 months of age, they have a very similar bacterial composition, but then at 1 year, and especially at 2 years and 7 years, they diverge a lot. So the oral bacteria present in children that were breastfed for a longer period of time actually is very different from the ones that were breastfed for shorter number of months. We have here an example. For example, we see here, for example, that in children that were breastfed for longer, the levels of porphyromonas were higher. Sorry, were lower. So the ones that were breastfed for a shorter period of time had higher levels of porphyromonas and also other oral pathogens. So we have to study this in more detail, but it is clear that there are some factors that actually have an influence, not only at the beginning, but in the long term. One of the reasons for why breast milk actually could have such a deep and long-term impact is due to the fact that breast milk contains many oral organisms which are going to be able to colonize the oral cavity. I don't have time to go into this, but this was the PhD thesis of Alba Boix, and she showed very clearly that some bacteria in breast milk are able to enter the oral cavity and to colonize. But at the same time, I want you to look at some of the compounds present in breast milk. And we have these human milk oligosaccharides. This is a specific kind of sugars that the baby is not able to digest. However, they are present there because they are nutrient source for oral bacteria and gut organisms. So that means that breast milk contains some sugars, not because they are good for the baby, but because they are good for the some of the organisms that have to be able to colonize the gut and the oral cavity and so on. Which this is what what we would call today a prebiotic, substance that when we incorporate it into the diet, it's going to favor the growth of some organisms which are good for our health. So this is why I say that human breast milk is a source of both prebiotics and probiotics, which is some live organisms that when we incorporate them in our diet are able to perform some good function for our health. So I think that human evolution already invented these two concepts, and breast milk is actually probably the first source of prebiotics and probiotics in the history of mankind. So that's probably one of the reasons why it's going to have a deep impact in the development of oral microbes. The other factors that we found had a very important impact was actually the use of antibiotics in the first 2 years of life. This image is a bit more messy, but what we see here is that at 2 and 7 years of age, individuals that had taken antibiotics, in the first 2 years of life, actually have a very divergent set of bacterial composition. So the composition of oral microbes is very different if you took antibiotics very early on in life. So in general, when we put together all this data, I now believe that the way that oral microbes are able to invade our oral cavity and to settle down follows what ecologists call an ecological succession. We have some organisms at the beginning that change a little bit the soil, they change a little bit the conditions. This allows other organisms to be able to settle down. And then with the years, a big forest is going to be formed, which is, the oral microbes that we found in an adult. And this underlies that it is very important what happens here at the beginning, because if you have a fire here, it's going to be easier for you to recover and go back, to have a final forest. And this is actually what happens as adults when we take antibiotics. When we take antibiotics, we're going to eliminate most of our microbes in the gut and in the oral cavity, but it has been shown that after 1 month or 1.5 months, we tend to recover the same organisms that we had before. That's because our immune system is already mature, it's already adapted, it knows which ones are, it's able to know already which organisms are ours. It's able to tell apart, our own cells from extraneous cells and so on. But in the very first year of life, this is a very important and very delicate period where we have to be exposed to the right organisms. And therefore, this is why when we take antibiotics here at the beginning, at this very fragile period, we may end up having not a pine forest, but perhaps an oak forest or something very different, because what happens at the beginning is going to condition, to have an influence of what happens afterwards. So this is why I think that we have to emphasize the oral health of the mothers during that first year of life, and have to expose the babies to the right set of organisms. Okay, apart from that, some of the work that we have been doing based on the DNA has been very important to unravel which are the organisms causing different oral diseases. If we focus, for example, on dental caries, and we take samples from the healthy dental plaque, and then from initial and more advanced dentin cavities, we have been able to identify which organisms are present. This was the PhD thesis of Aurea Simone a few years ago. And the more DNA in the cavities that she sequenced, the higher number of species that she found. And then you see that after sequencing a little bit, there is not a higher number of species, because we have actually been able to identify all organisms present in the sample. And the interesting thing here is that when we compare the number of bacterial species found in the healthy dental plaque, this is very high, over 500 species, like we said before. But when we look into the cavities, especially at the initial enamel caries lesions, the number of bacteria actually drops a lot. So from 500, it goes down to 100 species. This is still, of course, very high, but there is this drop as an indication of a more extreme acidic environment. Many bacteria are not able to survive under these low pH conditions, and therefore only a few species are able to survive and to expand into the cavity. But of course, 100 species is a lot. We had the idea in the past that dental caries was caused only by mutants Streptococci, but here we find over 100 species. But of course, when we first published this, people told us, well, perhaps many of these species that you identify based on the DNA are not active. They might even be dead. So you need to have some some method to be able to tell apart which bacteria are present from those which are active. So I always compare this with what happens, at least in Spain, in a construction work. We always say that if there are 5 people in a construction work, only one of them or 2 are working, and the others, are just chatting or looking at the others working. So if we extract the total DNA from here, we would think that all 5 were actually being present we're actually having a role, having a function in building the building. But we have a trick. If we extract the RNA, which is the active part of the of the cells, which genes are being expressed, which organisms are active at a particular time, then we will be able to identify those live organisms which are actively growing. And that's what we did, to extract the RNA and to sequence the RNA to obtain the metatranscriptome of cavities. And when we do this, we identify bacteria which are alive, which are active, and we again find a very high number of organisms in different cavities. So I have here, for example, 6 dentin cavities from different individuals, and we see each color is one particular genus. And as you can see, the composition of bacteria in different cavities is entirely different. This makes it very difficult to identify a single causing agent because there are many of them, and to make things more complex, the composition actually also varies between individuals. This is the most extreme case that we found because not only it varies between individuals, but also between cavities from the same individual and even from the same tooth. So we have here a molar, a 4-7 molar, where we have 2 cavities, an initial enamel caries lesion, and then a dentin cavity. We collected samples from them, and then when we sequence, we see that the composition of bacteria is also very different. So what can we extract from here? That actually having a vaccine against dental caries is going to be very hard because we don't have a single target. And for decades, many groups have been trying to develop a vaccine against dental caries. And what's the target that they have used? Well, the surface antigens of S mutants. And where is S mutants in here? I'm going to mark it here with my mouse. It's this less than 1% of the total of active organisms in cavities. And we found about between 40 and 50% of the individuals with cavities do not have S mutants. So it's not the only organism causing the disease. And when we have tried to develop passive and active immunization strategies, like having a vaccine against dental caries, then it goes into clinical trials and it doesn't work well. And this is because even if we are able to produce antibodies against S mutants, we still have 99% of bacteria able to produce acid and to contribute to expand a cavity. And that's why I think it's not a matter of getting rid of the biofilm. It's not a matter of having a vaccine. It's actually a matter of keeping a balance. And the same thing, I think, can be applied for gum diseases. So we have here the composition of some organisms from the pockets in people with gum disease. And we see here as the disease becomes worse, so the depth of the periodontal pockets is larger, we have an increase in some of the bacteria which we know are involved in the disease. We have the red complex bacteria and some other organisms like filifactor that we know are also involved. But I want you to look not at what happens here in the disease, but I want to look at what happens under healthy conditions. Individuals without pockets, if we look at the healthy gingival sulcus and we take a sample there, these pathogens are also present. But of course, they are only at 1% or even less than 1% of the total. But it doesn't mean that we actually get infected with these organisms. And that's why I believe that oral diseases are not the typical infectious diseases as it is still said in many textbooks. It's not a typical infectious disease like, Vibrio cholera, that you actually drink some water that has this organism and then you develop the disease. In this particular case, we all have these organisms. We all have the enemy at home, So what happens is not a matter of getting rid of these bacteria or cleaning extremely the biofilm to get rid of them. Actually, the main point is that they are going to be present and something happens that breaks that balance. Under healthy conditions, individuals without gum disease also have those pathogens, but they increase in proportion. So I think that's why I believe that we have to work on trying to keep the balance and instead of trying to get rid of those oral pathogens. That's why I believe that dental caries and gum diseases are not infectious diseases, but I think it's better to treat them as the product of a dysbiosis, that is, a breakdown in the balance between the host and the bacteria. So they are actually, we have seen already that they are highly polymicrobial, and they are actually caused by a dysbiosis, by a breakdown in the balance between the health so between the host and the organisms. And they are called not by oral pathogens, but it's actually probably better to call them pathobionts. That is, oral bacteria which are present also under healthy conditions, but at very low levels. And when the balance is broken, then they increase in proportion, causing the disease. So that's why I think we have to focus on strategies to restore the balance. That's why I believe that the use of prebiotics and probiotics is going to be very important, trying to restore that balance. And just to give some insights about how can we actually study better how to keep that balance, I'm going to show you at the end of my talk to mention the research that we did a few years ago comparing the metagenome, all the DNA sequences of the bacteria present in dental plaque from caries-free and caries-active individuals. What we did was to take dental plaque samples from these two groups of individuals, extract the total DNA, and then by high-throughput sequencing, we were able to identify not only who was there, but also which were the functions that they encode, what were they doing. And it was very interesting to see that some of the bacteria associated with health include Rothia, Neisseria, that is, organisms able to reduce nitrate into nitrite and nitric oxide. So they appear to be, they are always consistently found in individuals with good oral health. And it was very interesting to see also very high levels of what we appeared to be a new species of Streptococcus. So we decided to try to culture that organism, and we went down to those caries-free individuals until we were able to isolate this organism in pure culture. And we found that it was a new species that we call Streptococcus dentisani, which means healthy teeth in Latin. It's this beautiful organism here. And the really exciting moment came when we were able to culture this organism in the presence of the bacteria causing dental caries like S. mutans and others. We have here a lone culture of S. mutans, and in here in the middle we have our anti-caries organism. And as you can see, there is an inhibition ring around this colony, because the S. dentisani is able to produce some antimicrobial peptides which are able to dilute into the agar and prevent the growth of the pathogen. And we found the same here against other oral pathogens. And that it was extremely exciting, as you can imagine, in the lab, because we realized that caries-free individuals actually have some good organisms which are probably able to prevent the increasing proportion of oral pathogens. How do they do that? Well, this is what happens to S. mutans after being exposed for 15 minutes to the supernatant of S. dentisani. And I'm going to mark you here with the mouse. You see those little pores, these little holes here, here, here as well. So this is the typical, another hole here and here. This is the typical mode of action of antibacterial peptides, which is some kind of natural antibiotics which are produced by bacteria to fight each other. And what happens is that, after the pathogen has got all these holes which are produced by these antibacterial peptides, then the cell is not able to keep the osmotic pressure between the inside and the outside of the cell anymore. And then what happens is that it bursts and it lyses. And this is what we see here. These are the leftovers of S. mutans after being exposed to S. dentisani. And as you can see, it is releasing all the intracellular content. And this is the way that they die. So this is, we believe, is what is happening actually in the dental plaque of caries-free individuals because of the presence of these organisms which are able to protect us from oral pathogens. So when we look, for example, at the presence of this S. dentisani in different cultures, we see that it's not only present in Spain. We have got now data from Egypt, Chile, the US, and so on. And these are the last ones that we got in Mexico. And we see that people that have no caries, caries-free individuals have higher levels of S. dentisani. People with caries have much lower levels, and some of them actually have none. So we believe that this is a health-associated organism. And of course, we could use it as a probiotic, so the question is whether we can actually incorporate it into the oral cavity of people with cavities, with caries, to prevent the development of caries. So we have made a few clinical trials with this. The way that we actually put the S. dentisani, we have it in a powder, we mix it with water, and then after about 20 seconds, it becomes like a gel. And we apply it in these dental splints, dental the dental ferules for about 5 minutes per day. We have tried applying it one day, once per day for a week, or every other day for a month. And we see that a single application is enough to have some effect, but the best effect we find it when we apply it either for a week or 14 times in a month, once every other day for a month. And apart from having an effect on the amount of, having an effect on the pH, it has been also very interesting to see that in comparison with a placebo, we found a lower amount of plaque and a lower gingival index when we apply this organism in the oral cavity in comparison with a placebo. And we have found that this is because it's able to kill the bacteria making dental forming the biofilm like we saw before, because one of these antibacterial peptides that is able to produce is able to kill Fusobacterium, and we have found actually that this is the reason probably why the amount of plaque and gingival inflammation is lower in these individuals after we apply. So it has been very interesting, and we are now trying to develop it as to be put into chewing gum, yogurts, and so on. And we have been able to make the scale up to make it, and we hope that in the few years it could be in the market. And not only we can incorporate these organisms, but also some compounds which might favor the growth of health-associated organisms. And one of these could be nitrate, as we saw before. Nitrate can be transformed into nitrite and then into nitric oxide or ammonia. So we wanted to see whether this was actually adding nitrate into biofilms would be able to buffer the pH, and this is exactly what we found. So we see here in an oral biofilm model what happens with the nitrate 5 and 9 hours after we add it, and we see that at 5 hours most of the nitrate has been consumed, and then by 9 hours there are no nitrate left. This is because nitrite has been able to, the nitrate has been transformed into nitrite. So that's why we see an increase in nitrite after 5 hours. And then by 9 hours, the nitrite has also all been used up and has been transformed into ammonia or nitric oxide. So we measure the levels of ammonia. We see that they are higher at 5 and 9 hours in the samples that we have with nitrate added. And at the same time, when these enzymes transform nitrate into nitrite, they use lactate, because lactate is an electron donor for this chemical reaction, which means that not only is going to produce ammonia, which buffers the pH, but at the same time is going to consume the lactate, as we see here. So both at 5 and 9 hours, the amount of nitrate is lower in the group being treated with nitrate. So this means that because there is less lactic acid in the environment and higher levels of ammonia, the pH is going to be buffered. So in the absence of nitrate, we see that at 5 and 9 hours, the pH is going to drop, whereas in the presence of nitrate, the pH is going to be balanced, is going to be buffered, and therefore it doesn't go below 7. This means that nitrate is going to be probably a very good compound trying to balance the ecosystem and to reduce the acidogenicity of the plaque. But not only that, if instead of making ammonia is going to produce nitric oxide, which happens when the pH is a bit more is not acidic, and in the lower presence of carbon and higher levels of proteins, then what happens is that it's able to produce nitric oxide, and that's going to be very beneficial for killing gum disease pathogens. We have been able to isolate some of these bacteria. We have agar plates with a compound that changes color into this beautiful red color when there is nitrite in the medium. And therefore, we have been able to isolate some of these bacteria that we intend to develop as health-associated organisms. And this is what happens when we add the nitrate into an oral biofilm. We have some organisms which increase in proportion at 5 and 9 hours after adding the nitrate. So these are the bacteria able to consume the nitrate and to transform into nitrite and into nitric oxide. And then we have these organisms that go down in proportion as a consequence of that. I have marked here in red, with these 3 dots, the 3 members of the red complex of gum diseases. So it's able to reduce the numbers of these and many other organisms also involved in gum diseases. So we believe that when it's able to produce nitric oxide, many of these organisms causing gum disease, gingivitis, gum bleeding, and so on, are actually strict anaerobes, and they are very sensitive to nitric oxide. So when we add nitrate into the system, these health-associated organisms are going to be able to produce nitric oxide, and that's going to kill strict anaerobes, which are involved in gum diseases. So that's why we believe that addition of nitrate, for example, in the diet, or even nitrate in a toothpaste, is going to be very good to improve our general oral health. So in conclusion, dental caries and gum diseases are polymicrobial. So they are probably not the product of a single species, but a group of species that have a synergy causing the disease, and they are not the product of an infection. We see them more as a product of a dysbiosis, of a breakdown in the balance. And some individuals appear to be very resilient to change, and we can learn a lot from them. So we can see which organisms are present in those individuals, what do they eat, and so on. We believe now that the caries vaccine may not be effective because we don't have a single target. We have multiple species, and it's going to be impossible to eliminate the biofilm. That we can try to modulate it so it goes in the right way, increasing the levels of health-associated and decreasing the levels of pathogens. We can do that by promoting our natural defenses. For example, increase the flow of saliva, and having the right compounds in the diet that are going to stimulate the growth of health-associated organisms. And we can help that by using some prebiotics, for example, arginine, which is going to help to keep the balance in individuals with dental caries, or nitrate that we believe are also going to be good to keep the balance in people with gum diseases. And of course, not only adding the compounds to stimulate the growth of health-associated organisms, but also we can actually add these organisms in the form of probiotics. And not only as Dentisani, there are many other interesting projects around the world, like the Streptococcus A12, which has a very high arginolytic levels, and is going. So in the next few years, I think we are going to see all these new compounds and products being present in the market and available to improve our oral health. So in general, just a general message to end, like in this book by Martin Blaser, where he actually talks about many organisms that we are losing due to the modern way of life, including the overuse of antibiotics and so on. We have to look at bacteria, not only at those bacteria causing the disease, but also looking at those bacteria which are able to protect us from disease. We need to keep a balance. We need to actually take care of our good oral bacteria, because they are also play an important role in keeping a healthy balance in our oral ecosystem. So let me just thank my team. Many of them are no longer with me. They are doing postdocs in other countries, and I wish them all the best. And it has been a very big pleasure to work with them. And I hope that in the near future, some of the results that we have actually found can be applied into the real world, because I think they have very important clinical implications. So thank you very much for the invitation, and I'll be happy to answer any questions that you might have. Sorry, I cannot hear you.
Yeah.
Okay, no.
Okay. Can you throw some light on the connection between the oral cavity and the gut with regard to dysbiosis in the gut caused by oral microorganisms?
Yeah, it is a very good point, actually, because what happens is that oral organisms are very good in the oral cavity, but when they go outside the oral cavity, they start to cause a lot of trouble. So we swallow about 1 liter of, close to 1 liter of saliva per day, and so that's one way by which oral organisms can go into the gut. But I think the main route is now thought to be actually through the blood. So if you have gum bleeding and so on, then you have their hole open into the bloodstream, and many organisms can go from the oral cavity to the bloodstream and from there to other parts of the body. In general, we have very few oral organisms in the gut, unless there is a bad oral health or unless the patient is immune depressed. And when that happens, then many oral organisms appear to be also present in the gut.
Thank you. Do you think more the diversity, better would be the health of the oral cavity?
Well, in general, it depends. For example, in dental plaque, we see that the number of organisms actually go down in cavities, and that's probably because the environment is more extreme. So in general, also in the gut, it is considered that a high number of species actually equals, a higher level of species actually means better health. And that in general is true in most cases, except in gum diseases. When you have gum bleeding and so on, what happens actually is that there is an increase in the number of species in the pockets. And this is probably explained because the immune system at the local level in the gums is a bit changed, and that means that it's not going to be so efficient at keeping aside the pathogens. So, for example, we have an immune system altered by the action of P. gingivalis, and that means that many oral organisms which were not present, suddenly they are able to invade and to colonize. So under gum disease and inside the pockets, actually, we see a higher number of organisms, and that's because the immune system is not working properly. So in general, a high number of species means more health in most parts of the human body, but in particular in the periodontal pockets, this is the other way around.
Okay, thank you. What about the lifestyle factors contributing to dysbiosis?
Well, very important. I think that when we look at oral diseases, we cannot look only at the organisms alone. There are 3 main features involved: which organisms you have, then the internal features of the host, your genetics, your immune system, the amount of saliva that you produce, and so on. But then we also have the external factors, mainly the diet, oral hygiene, and so on. And it's only when those 3 factors come into place at the same time, the wrong set of organisms in a host which is predisposed, and then with external factors like the diet and oral hygiene and so on, all of them are going to come into place, and then we have a perfect storm where the disease is going to be formed. So I think the external factors are really important to be taken into account. Diet is very important. Smoking is also very important as well. It's going to have an influence on the immune system in the mucosa, and that's going to also have a factor, especially for gum diseases. And the oral hygiene is also very important. For example, the higher amount of plaque that we have, the higher level of inflammation, and the more plaque that we have, the lower the pH is going to get lower as well. So after having a meal, the pH is going to drop. If we have just brushed our teeth and we have no plaque, the pH is going to drop very little. But if we don't brush our teeth for 12 hours, 1 day or 2 days, then the pH drop is going to be much larger. So less oral hygiene, it means a larger amount of plaque, and this implies a larger drop, so more risk of having caries and also more inflammation.
Okay, thank you. You have talked of dysbiosis. So do we have the concept of rebiosis?
Rebiosis, I like that. I think so. I think we can work on trying to restore the balance. I really believe that that's possible. And that's why I think that, having changes in your oral hygiene, in your diet, in the kind of toothpaste that you use and so on are actually things that can revert this process and go back to having a healthy, balanced ecosystem. And I also believe in the concept of what we call eubiosis. So even if you don't have a disease, you can actually work on trying to improve your oral health to be more resilient to change. Actually, you see, for example, that people that eat lots of people that eat lots of vegetables actually have a higher levels of these health-associated organisms, like the ones that are able to transform nitrate into nitrite. And that's because we ingest more than 80% of the nitrate in our diet due to vegetables and fruits. And therefore, people eating more vegetables and less meat are going to have a higher levels of these organisms, because the nitrate in the vegetables are going to promote the growth of these health-associated individuals. So I do think also that we can revert the process of the dysbiosis, and at the same time we can improve a general state of health, improving this eubiosis, by changing our conditions, our diet, our oral hygiene, and so on, trying to improve further our oral cavity and trying to drive that balance into the right end.
You have spoken about this role of nitrates. You have spoken about role of arginine. You have spoken about some prebiotic bacteria that would help in restoring homeostasis. Do you see any role for nanoparticles in facilitating establishment of a healthy flora? Because a lot of anti-plaque strategies are directed using nanoparticles. So,
right. Yeah, thanks for that question. I do believe in that, actually, because when we, of course, I don't like, for example, the use of oral mouthwash on a daily basis, because I think it is not good actually to remove all the organisms in the oral cavity. At some particular points, it is important during the treatment, but not on a daily basis as part of your daily oral hygiene. And that's because you are going to get rid not only of the bad organisms, but also the good ones. So I think that's why I think that the use of nanoparticles, which are a bit more intelligent, are actually going to be better, because they only act under the right set of cases. For example, there are some nanoparticles that only open and release the content when the pH is acidic. So that's why I said that they are in a way more intelligent, because you could have antiseptics inside those nanoparticles, but they are only going to open if the pH is very acidic or if the pH is very alkaline. So they are only going to perform their action under the conditions that we want them to do, but not all the time. So I think this could be a very good application of nanoparticles. I also think that they could be very interesting in the trying to disinfect the root canals in endodontic infections. I think that over 10% of the endodontic treatments do not work well, and that's because it's very difficult to sterilize all the channels and the accessory channels and the root canal and so on, because of the shape and so on. Whereas if we actually are able to put their nanoparticles with movement, they will be able to move along the channels and release the antibiotic or the antiseptic as they go through those channels. So I think that's also a very promising strategy, try to disinfect the channels in endodontic treatment. So I do think that in the future, we are going to see more and more of these nanoparticles being applied in different kind of treatments.
Thank you. There's one more question, but may not be directly related to your talk. Any work in progress related to oral health and COVID-19 in your lab?
Yes, well, we have just finished the clinical trial trying to see the effect of oral mouthwash, trying to reduce the load of the SARS-CoV-2. We now know, that the SARS-CoV-2 actually attached to ACE2 proteins which are very common in the oral cavity. In fact, they are extremely common in the tongue, which can be actually a site for attachment of the virus. So actually, the levels of the virus can be very high in the oral cavity before invading other parts of the body, like the lungs. So it would be very important to have a way to reduce the viral load. And we have tested 4 different mouthwashes in a clinical trial, in a randomized double-blind clinical trial. So far, we have seen that the different mouthwashes, although they work very well in vitro, in vivo we see that there is not a lower viral load after the use of these oral mouthwashes, although this has been measured by PCR. So what we are doing now is actually to culture those viral particles which are still present to see whether they are able to infect or not. So we will know more in about a month or so. So we still cannot say actually that an oral rinse with an antiseptic mouthwash is able to kill the SARS-CoV-2 virus. And the other important thing that we are not working on, but I think has been very important in the field, is to show actually that people with gum disease have a worse outcome of the disease. You know, they have a better COVID-19 outcome when the oral health is worsened. And it seems that this could be due because of the presence of periodontal pathogens actually increase the amount of inflammatory molecules and cytokines and so on in the bloodstream, and that inflammatory state is going to make a worse outcome of the COVID-19 disease. So this actually links very well gum health and oral health with the outcome of COVID-19, and I think more research should be done along these lines. And again, I think this again indicates how important oral health is for general health in the body. I think it's something that we have to transmit to our patients and also to the policymakers, how important oral health is for the general systemic health.
There's a question related to whether fungi play a role in causing dental caries in children.
Sorry, can you say again? I didn't get it.
Okay. Is there a role for fungi in causation of dental caries in children?
Well, very good question. I mean, there are some indication that Candida is also highly acidogenic and can be involved. So yes, there's probably a role, but it's not my field of expertise. I think that we have to study the interaction between fungi and oral bacteria. I think that we are making the mistake of actually studying either fungi or oral bacteria, and there are very few studies where both of them are, studied at the same time. There is some evidence that they could actually have a synergistic effect, and I think even though the levels of Candida and so on in cavities is much lower than that of oral pathogens of the bacteria, I think this is a bit underexplored and understudied. So I cannot tell you how important it is. I know that it plays a role, and I think that more research should be done, not only to study the presence of Candida, but also the interaction with the bacteria also being acidogenic. There are specific interactions by which some oral bacteria attach to the surface of Candida. So I think that there is more there in those interactions to study that we still don't fully understand.
Thank you. That's all we have. Thank you very much for this very informative lecture. I now request Dr. Faizuddin sir to hand over a certificate of appreciation to Dr. Meera.
Thank you.
It gives me immense pleasure to hand over this certificate of appreciation on behalf of Rajiv Gandhi University of Health Sciences, on behalf of Bangalore Academy of Periodontology, and on behalf of VS Dental College.
Shriyansh, can we have the certificate, please?
Thank you so much. I really appreciate it.
Yes.
Thanks for your contribution. Thank you very much.
thanks to you. It has been an immense pleasure and a big honor. Thank you so much.
Welcome.
I now call upon the next speaker of the symposium, Professor Ramaj. It's my pleasure to introduce him. Professor Ramaj graduated from Edinburgh University with a BSc in Medical Microbiology in 1996. He then received a PhD from the Queen's University of Belfast in prosthetic joint infection in 1999. He subsequently undertook 2 postdoctoral research positions over a 4-year period, focusing on fungal and bacterial biofilm infections at the University of Texas Health Science Center at San Antonio and University of Calgary, Canada. In 2004, he returned to UK and was appointed as a lecturer in microbiology at Glasgow Caledonian University Glasgow Caledonian University prior to taking up the position of senior lecturer in microbiology at the University of Glasgow Dental School in 2007. Professor Ramaj has been the head of the Biomedical and Medical Sciences and the lead for Infection and Immunity Research Group since 2019. Professor Ramaj received his personal chair in 2012 and was promoted to Professor of Medical Microbiology. He is the Honorary Secretary of the British Society for Medical Mycology and acts as a consultant and key opinion leader for a number of industrial partners. Professor Ramaj has continued to work in the field of clinical biofilm research, both bacterial and fungal, since joining the department. The primary focus of the work undertaken within Glasgow Dental School and through international collaborations is in the treatment and resistance of microbial biofilms. Under his direction, the group is actively involved in collaborative projects with GlaxoSmithKline and other multinational industrial partners to establish and develop novel biofilm models that reflect both bacterial and fungal biofilms of the oral cavity, respiratory tract, and biomedical devices, which can be utilized for small molecule research. Welcome you, sir, and over to you for your lecture.
Okay, thank you very much for the introduction. So it's a great honor for me to talk to you today. It's very difficult to follow Alex Mira, an excellent talk, and then thankfully the last question that was posed is a nice segue for my presentation. My presentation is going to be somewhat technical and preclinical, but hopefully for some of you listening, then maybe you can take some inspiration on what's possible in order to evaluate and test things out in a laboratory that are relevant to oral health. So the title of my presentation is there a need for complex biofilm models in oral disease? And this is going to be a sort of personal journey of working with industry, and in particular GlaxoSmithKline. So what I'm going to tell you a little bit about is my understanding of what I think about in terms of what biofilms are. Tell you a little bit model systems, and then I'll go into a bit more detail about some of these models and how we've developed them, some of the caveats of them, how we analyze them. And then I'm going to introduce the importance of the keystone pathogen, and don't get excited because it's perio. It's not P. gingivalis, it's actually Candida albicans. And then I'm going to talk a little bit about some of the microbiome studies that we're doing or done, and using those to try and inform and improve our biofilm models. So that's what we're going to go through over the next 45 minutes or so. So I've already had an introduction in terms of what I am, but I like to just give my sort of brief biogeography of what I've done. So just to give you an understanding of who I am and what I do. So I did my PhD on prosthetic joint infection, so in Northern Ireland, which is just across the sea from where we are in Glasgow. And that's where I got an interest on bugs sticking to surfaces. Okay. Then I went over to San Antonio in Texas, where it's much better climate, probably more like India than Scotland at the moment. It's raining and very cold. But there I grew an interest on Candida and yeasts and how they stuck to surfaces, both in the oral cavity, but principally in terms of catheters and biomedical devices. In order to improve or diversify my portfolio, I moved up to University of Calgary, and there I worked on organisms like Pseudomonas aeruginosa and Burkholderia senescipacea in context of the cystic fibrosis lung. And then back to Glasgow to the dental school. And since then, although I'm going to be talking about dental research, then a lot of the work that I do is related to respiratory tract infections, GI, urinary tract infections, medical devices, wounds, a whole raft of things. So my ultimate interest is in how bacteria and fungi stick to surfaces and how, by sticking to a surface, that changes the way in which we can manage them. So to start with, what is a biofilm? So in essence, it's a matrix enclosed microbial population which is adherent to each other and or to surfaces or interfaces. So I think the important thing to take there is it's not just about bacteria sticking to a specific surface. They can attach to one another as aggregates or clusters. And this idea that they're enclosed by some sort of matrix, some sort of polymer, some glucans, polysaccharides, so on and so forth. These things are really critical in terms of the adhesive capacity, but also in terms of stopping things from getting near the microbes, particularly the antimicrobials and host immune cells. And this is what you'll see probably in any given textbook. So you have bacteria which attach to a surface, they start to lay down this polymer, they get these mushroom-type shaped colonies. I want you to dispel that kind of image from your head. This mushroom-shaped kind of colony, these long towers, are only really representative of Pseudomonas aeruginosa, one particular type of strain under one particular type of condition. Yet, for some reason, it's penetrated into the larger, standard textbooks for medical students, nurses, dentists, and beyond. It's fairly artificial. You can see here that this particular biofilm doesn't look like that at all. Yes, there's microcolonies, but we don't have these large towering structures at all. And you'll see we'll come back to these sort of multi-species organisms. And this fits the definition of a biofilm as well. The red cell here is Candida, and you can see these Streptococcus attaching quite nicely to it. That, to me, is also a biofilm because you've got these organisms forming a biofilm on Candida. Okay. Biofilms are indeed ubiquitous. So yes, as dentists or people working in the dental field, then we're interested in teeth, but drinking water, manufacturing, oil recovery, ships' hulls, so on and so forth, obviously medical implants, all these things and all these different materials that are in aqueous environments or in some sort of water-air interface have the capacity for microorganisms to stick. So you'll find biofilms pretty much everywhere. In terms of the human body, then, here's Da Vinci's Bionic Man, and you can see from the plethora of different examples here that as soon as you have an implanted material, then that gives a microorganism, an endogenous microorganism, usually the opportunity to stick to that surface. And when we think about it in terms of the oral cavity, then pretty much every disease which is microbially associated is a biofilm, whether it's caries, gingivitis, orthodontic appliances, endodontics, angular colitis, denture stomatitis, so on. So all these things are associated with bacteria and fungi. And as Alex has alluded to in the previous presentation, then there's a whole raft of microorganisms, and we're only really just getting to grips with what those look like. There's a number of microbiome studies that have been performed since 2010 onwards, moving on from 16S cloning technology. And the more and more of these studies are done, the better clarity we're getting on what causes different organisms. But nonetheless, there's a whole raft of organisms of differential importance associated with these diseases. So in terms of biofilms in the oral cavity, I guess, what is there? What are they doing? How do we model them? And what techniques do we use to investigate them? So, I mean, Alex touched on this as well. So, I mean, this is kind of where we used to be. This is a paper which Jason Brown from our group led. So some really nice figures, which I'll show you throughout this presentation. But essentially, this is where we were, pre-60s and onwards. We take conventional microbiological samples from the oral cavity. We put them on plates. We might do antimicrobial sensitivities, and occasionally we might look under the microscope. And arguably, I'm not sure what it is in your individual institutes, but very rarely do people send samples off for microbial identification, because ultimately you're going to treat these patients empirically. It's those patients that aren't maybe responding that you might want to find out a bit more specifically what they do. But, we should appreciate that these oral biofilms are complex. So targeting one organism is actually pretty difficult and possibly pointless. But in terms of the way we study the organisms, it's still really important from a research point of view to know the types of organisms we have, how they respond to antimicrobials. Moving on, we were able to then take these organisms from plates and then create these flow type systems. We were able to use anaerobic microbiology and electron microscopy, moving on to confocal microscopy to look at them, to see how they appear down the microscope. The group in Calgary created the Calgary biofilm device, which is sort of a peg plate model you can see here, which has got its nuances. But a lot of people use these systems where you grow organisms in the bottom of a 96 well plate. Growing biofilms is pretty much, you can grow biofilms on whatever surface you want to. You've just got to be a little creative. But what we have been able to do over the last 20, 30 years is to actually analyze them in a little bit more detail, rather than just throwing a single organism in, looking how it sticks to a surface, and then counting it using standard plate counting methods. We can take a molecular approach. We can extract the DNA or the RNA, and we can determine what it's doing, how much of it's there, and we can even sort of take these samples directly from the patient's mouth and look at more complex biofilms, or we can put them into systems which allow us to look at it with immune cells. So I'll kind of go through some of these different approaches when I'm talking about some of the studies that we've done. So this is, for me, the biggest issue or failing of sort of oral microbiology, and a lot of other disciplines in applied microbiology, is the fact that there's a propensity for researchers to look at their favourite organism, whether it's Streptococcus mutans, Porphyromonas gingivalis, Candida albicans, Staph aureus. There is a propensity to hone in your own organism and grow it in a single species environment. We can take knowledge from monoculture in terms of agriculture and the problems that occurs, because it's much more sensitive to change. It's not a natural environment, therefore perturbations of this system, it's much easier to perturb a monospecies environment, whereas what we want to try and do is create new antimicrobials or new strategies to look at oral biofilms in a much more realistic fashion. Obviously, taking complex plaque samples from individuals, the reliability and reproducibility of those analyses is difficult. So what we need to try and consider is a halfway house. But in saying that, that's where most of us started off in monoculture. So if you're screening for a new active, for example, or screening an existing active, a mouthwash, let's say, for example, a mouthwash, chlorhexidine, or Listerine, something like that, then this is the standard way in which we would do it. We take our organisms from the freezer, we put them on a plate, we make sure there's a pure colony, we grow them up overnight, we spin them down and wash them, and then we're able to count them either in a hemocytometer if you've got something bigger like a yeast, or a spectrophotometer for bacteria. And then you're going to put it on a 96 well plate of some sort and test the drug. And that can either be planktonically, meaning free floating. And free floating microorganisms in the context of the oral cavity is maybe relevant to saliva, but nothing else. So you're not really going to be wanting to give an antimicrobial just to kill bugs in the saliva, because most of the organisms you want to manage are sticking to a surface. Therefore, getting an appropriate substrate, putting the bacteria on that substrate to grow, and then giving an appropriate drug. That's kind of the conventional way in which we would think about treating, and that's kind of the bread and butter of what our laboratories would do to a certain extent. So I mentioned I'm going to talk about my personal journey in terms of GSK. So I came to the dental school in Glasgow in 2007, and a lot of the background I wanted to do was in Candida albicans from the work that I'd initiated in Texas and San Antonio. So prior to taking up the position in Glasgow, I put in an application for the IEDR GSK fellowship, and between getting the job in Glasgow University and arriving, then I actually got the word that I'd got this award. So one of the things we wanted to do was to look at naturals, and one of the things we did, along with Professor Culshaw, who's going to speak afterwards, is look at tea tree oil. So there's lots of interest in naturals. There's so many different unexplored molecules out in nature, rather than conventional things like azoles. We know that fluconazole, voriconazole, and so on are actually completely ineffective against Candida biofilms. So using a technique which I devised with my group in Texas, which is called XTT, it's a very, very simple assay. And what it is this colourless tetrazolium salt. And if you add this electron coupler imidodione in the presence of an active viable cell, then it reduces this tetrazolium salt to this molecule here, the formazan, which is orange. So if you have live cells, they appear orange. If you've got inactive cells, they're clear. So it's a very, very simple assay. And basically, you can see here this depiction here, untreated. Here, you've got a high level of absorbance, which is supposed to be the orange, whereas your media only, there's nothing, and there's different drugs, you get a differential response. So using that type of approach in a monoculture, we were able to screen a whole range of isolates from cancer patients to look and see whether tea tree and its derivative components could be effective. So I'm showing you this really just to show the simplicity of an assay which can be used to fish or mine for different antimicrobial products. So there's a raft of things you can do. So it's really simply about taking a standardized culture of Candida, putting it on these plates, and then testing your molecular product. So it's effective. It leads to some useful papers, and it leads to some citations, and that leads to other things. And indeed, we used a simple model, you can see it here in live action, to sort of look at sort of denture cleansers, look at commercial mouthwashes, and even some new novel things called fulvic acid. So by using this simple approach for the first probably 3 or 4 years at the dental school, this is where we were playing. We were playing with Candida albicans, and we were using our knowledge from conventional antifungals, working in sort of more general medicine environment, and then using these type of approaches to show that you can effectively measure how effective over-the-counter mouthwashes and denture cleansers are, and that provides the opportunity to say, okay, there's deficiencies, and if there's deficiencies in what the general public are using to manage their oral care, then how can you improve that? Okay. But hopefully from listening to Alex, then you'll realise, and Alex uses the same analogies as I do, I think, because I'm very much in touch with thinking about ecology, nature, and the external environment in terms of how I think about biofilms. And this is how I think about the oral cavity. It's not that monoculture idea of a single organism that's dominant. It's a spatially arranged number of microorganisms that adapt to the space in which they're in. They have the ability to sense one another. Obviously, there's shade here from this tree, which then have an impact on what can grow around it. But nonetheless, it's an organised environment, and I think that's how you should be thinking about it. I think Alex used the idea that it's a rainforest, and again, I absolutely get that analogy as well. But so some sort of standard ecological environment where you have a whole diverse range of species living together. And a kind of, the work that has come before us, Sokransky, Collenbrander, so on, these models exist. And I think that's kind of interesting. There's been lots of studies on co-aggregation of different bacteria. And I think Alex demonstrated this sort of Fusobacterium nucleatum and the importance, of targeting that key organism. If that holds things together, then it's simple enough to think about. If you remove that, then that's going to, the tower's going to topple. So I get that. But the thing to take from this is that there's lots of different things going on. There's lots of different microorganisms. And if you're thinking about building biofilm models, then it's not as simple as just putting that together, because it takes a little bit of technical expertise. Just to touch on the point, biofilm development is not binary. Okay, so when I think about a biofilm, this is our planktonic cell, if you like. So this is our bacteria or our fungi. He's got a bike, so he's mobile. He's got a fishing rod, therefore he gets access to food, highly susceptible to the environment. So it makes much more logical sense for the organisms to get together, put down roots, create this city of biofilm encased in the matrix of concrete. But it just doesn't simply go from planktonic to biofilm. There is a developmental phase, and you can see here, this is Staph aureus. But nonetheless, I showed you the previous diagram of the mushroom, which I told you to ignore. This is a much more evocative and realistic idea of what happens in terms of biofilm development, where you get the cells, you get EPS, but importantly, you're getting these tolerant cells. So I think the misnomer really is that biofilms are resistant. Biofilms aren't resistant, they're just more tolerant. Resistance would probably be much more akin to thinking about a change at a genetic level, such as the MECA gene in Staph aureus, where it becomes MRSA. These organisms in the biofilm are highly tolerant, and not only tolerant, they create these aggregates, and these aggregates that either disperse from the surface attached biofilm, those still have the same phenotype, so they're still tolerant aggregates. So that's kind of what we think about. It's not just a switching from a planktonic cell to a biofilm. It's a spectrum of different developmental phases, and across that developmental phase, that'll be differentially sensitive to antimicrobials depending on its physical environment. So it's an active developmental process, and I think it's been touched upon in terms of it's a sequential process. So I'm going to use Lego as the analogy here, but for anyone who's played with Lego, if you were to take your tub of Lego, I've got 2 small children and I have lots of tubs of Lego. If you throw that tub of Lego down on the floor, you don't suddenly get a structure. You have to physically put that structure together. It'd be a chance event that you threw this down and got something like this. So we have to use that very same approach when we put these biofilms together. So I'm using a diagram from the head of the dental school in Glasgow, Professor Bagg's textbook, but it shows quite nicely that idea that you get succession. So this is your clean surface, if you like, the Lego base. It's got pellicle formation, so maybe that's the little dots. But you need the right bits of Lego to go down first. So you need the streps to be there first. They start to put down a little bit of glue, which then provides the access for the other organisms, other aerobic organisms. And once they've sort of taken root, then that might drive the oxygen down to become more appropriate for capnophiles, CO2 loving organisms, and then anaerobes. So you need that sequential process. And by the same logic, then we need to do that when we grow complex biofilms. So Professor Culshaw and I talked to GSK, and we got money for a BBSRC studentship, or sorry, just a GSK studentship at this point. And the idea was, you know, we'd learn from what we'd done with Candida, recognized, well, actually, Shauna said, actually, perio is important, and it doesn't involve Candida, but can we maybe grow a 4-species biofilm or do something a bit more exciting? I suppose that's the reality of it. So we did, and we looked at the literature, and we looked at some of those models, and we selected 4 organisms to try and grow together. And what we did through trial and error is we took strep mitis as our colon, or a sort of pioneer organism. We let that grow for a day. Then we add Fusobacterium nucleatum, and then we added the sort of quintessential red complex organisms, P. gingi and AA, along to this S. mitis fuso mixed biofilm. And then we added more artificial saliva, and then by day 7 we had our organism. So unlike the Candida albicans biofilm, which you can grow in 24 hours and do some testing, the lag time here is the time taken to put these organisms together. So if you take those 4 organisms together and try and put them and grow a biofilm, it just doesn't happen. You know, it doesn't grow in the same way. The organisms that grow are poor. However, if you build it sequentially, then you get an end product. So hopefully you can see this. So this is published in the Journal of Periodontal Research in 2016. But you can see P. gingi doesn't form any biofilm at all. There's a few cells dotted around. Fusobacterium nucleatum has got the capacity to form monospecies biofilms. A is pretty poor. And strep mitis, you see all these, well, maybe hopefully see some, but there's a fairly broad coverage of strep mitis. So this is a higher magnification. You can see these. Okay, so you start to see these big microcolonies. F. nucleatum seems to be the dominant organism. However, if you put them together, then you can see, you know, hopefully side by side, that these mixed species biofilms are numerically more. So you've got 10 to the 7 organisms, up to 10 to the 8 of particular organisms like F. nuc, whereas in the monospecies biofilm, over time, P. gingivalis is only about 10 to the 4, so 3 logs difference. F. nucs even 10 to the 6, so on and so forth, and strep mitis tends to go up. So what you see is that the organisms start to cooperate with one another, and you create this biofilm structure. And that's important because you can then use that to ask meaningful questions. And one of the meaningful questions we asked was in terms of what does it do to the inflammatory response. So we came up with this cell culture insert system. So these are transwell systems that fit into a 24-well plate. And what we were able to do was secure a cover slip at the bottom of this transwell with the biofilm on it and then invert it. So then we were able to bring that biofilm down close to the epithelial cell layer and then see what happens in terms of inflammation. And what we see is with a strep mitis monospecies biofilm, then there's a little bit of gene expression. So this is a panel of different inflammatory mediators. IL-8's upregulated 24 hours, but pretty much nothing else is. And you can see there's a difference. If you fix the biofilms, i.e. sort of heat kill or formalin fix them, then there's no inflammatory response. With the P. gingivalis biofilm, there's a little bit more activity. So P. gingivalis is a bit more pro-inflammatory than the strep mitis, as you would probably predict. But more interestingly, with I've coloured this orange, so this green for health, red for sort of pathogen, and orange for sort of somewhere in between, because you've kind of got a buffering effect of the organisms, but you see a much greater inflammatory response of other mediators. And again, much more response with the live biofilm. So the multi-species biofilm is much more pro-inflammatory than, for example, a monospecies biofilm. There is caveats in terms of cell numbers and so on, and we've tried to account for that, but nonetheless, we see that effect. And if you do a principal component analysis of comparing these monospecies to the live, either fixed, the mixed biofilm, both fixed and live, what you see is a different cluster. So you can see that the multi-species biofilms have a different cluster from the single species biofilms. So there's certainly a difference in how the immune response works there. And interestingly, when you look at the live biofilm, you can see up here that in the live biofilm, you've seen all this IL-8 expression at RNA level. However, in the live biofilm, you're seeing that depletion. And that suggests that the gingipanes, that the organisms within the biofilm, probably P. gingivalis, has been able to chew up the IL-8 and make it, so basically it's protecting itself from the IL-8. So what we have in essence is a system that's complex, and you're able to ask a question, at a inflammatory level, as well as in terms of looking for actives. So again, Emma Millhouse came after Anto on a BBSRC funded studentship along, again, supported by GSK. And we looked at the same model with respect to testing things like chlorhexidine, but also resveratrol. So basically, that's a polyphenol. So the idea was, the literature suggested that it had an antibacterial effect, but also an immunomodulatory effect. It does have an antibacterial effect on monospecies or planktonic cells, but not within the biofilm. You can see, compared to the biofilm, using simple viability assay, there's no difference. Chlorhexidine, as you can imagine, is highly effective. Interestingly, though, when you look at it with regards to, if you focus here, when we look at IL-8 as an output, then it's very low, suggesting that actually when you treat the same epithelial cells from the same model as in the previous slide, that you get this reduction in IL-8 outputs, suggesting that there is indeed an immunomodulatory effect. So basically, these biofilms again have been stimulated with the biofilm, both fixed and live. So you're kind of seeing these effects. And again, we've used this a model and applied it. This is Mohammed Shahzad, one of our. He was a dentist who came and did a PhD with us, but he was interested in polyphenols. So we screened a whole bunch of things, and things like curcumin were shown to be really important. But again, the point really is that you've got this 4-species model, which is obviously better than a monospecies model, and we've been able to apply it as an inflammatory output mediator to ask questions in terms of inflammation, but also to screen actives. So we wanted to then take that a step further, because obviously the critique would be that you've only got 4 species in there, and what does that look like? So Emma's project was to then expand that. So we added more streps into our initial health-associated biofilm, if you like. We had a whole range of other organisms that are associated with Sokransky's orange complex. So we've got another sort of subspecies of F nucleatum, Anisylin, so that's a misspell there, and Vienella dispar. So we've got more organisms in there, and then Prevotella is in A. So what we've done is we've upped the ante. We've gone from the 4 species to a 3 species, a 7 species, and a 10 species. And by doing the same sort of procedure, i.e. we use our pioneers, on day 3 and 4, we're going to add these organisms, and in day 5 and 6, we're going to use these. So instead of the 7 days to do the 4 species biofilm, this is now taken as 10 days. But the good thing was, in both models, we can then store those in the freezer, so we can freeze them down and then repopulate them. So these boxes represent what the initial model was. So you can see how we've been able to take what we've learned from the initial PhD and then expand that to have a model which is a step above what we were working with. So this was sort of the essence, is that this 3 species was our sort of a health-associated, our 7 species, which was more akin to gingivitis, so something sort of pro-inflammatory. And then when we put the sort of the red complex organisms, this was sort of our model, which is depicting periodontal disease. So what we're seeing is a model in which we can think about moving from health to disease. So Shauna and Jason supported this work and drove it. And what we've been able to characterize that. So we've added dimension of what we've been able to do. To differentiate streps using PCR is quite difficult given the conserved nature of streps. So it's difficult to pick the species apart. So we don't differentiate them there using PCR, but we can see that obviously you've put streps in, you get streps out. In the 7 species biofilm, you can see that it's dominated by Anis alundi. And then in the 10 species biofilm, then it starts to look slightly different. And we can look at that by scanning EM, transmission EM, and confocal. So hopefully by looking at that, you can see quite a stark difference. So you can build complexity to the biofilm model in a sequential manner. So then what we've done is we've taken that model and then attempted to ask similar questions in terms of inflammation, but rather than look at it specifically on a monolayer of epithelial cells, we've taken a skin-ethic model, a human gingival epithelium, which has the keratinized layer and is much more physiologically like the oral cavity than a monolayer, and started to look at inflammatory outputs. So again, we've got gene expression and we've got protein. And what we see is that if you look at the filtered supernatants from these biofilms, whether they're 3 species, 7 species, and 10 species, then there's little difference. The unfiltered, you start to see differences. So there's clearly molecules or proteins released that are pro-inflammatory. So this is the 10 species and the 7 species. So you're seeing this increase in IL-8, and the biofilms are more pro-inflammatory. Okay, and again at the protein level, you're seeing not an awful lot here with the filtered supernatants. Interestingly, the unfiltered supernatants, you're seeing that elevated IL-8 with the 7 species, which is followed through with the biofilm. So what we've got is a 7 species biofilm, which is actually much more inflammatory than the 10 species, suggesting that again, this is hypothesis that the red complex organisms are modulating that immune response. And you can look at it at a whole range of other molecules as well, using sort of cytokine arrays, PCR arrays. But you're seeing again the 7 and 10 species. So we've got a system that you've got health and different sort of levels of disease system that you can interrogate your inflammatory question. So just to summarize that, so hopefully that's demonstrated that oral biofilms are complex. And I think it's absolutely clear that you need to use sequential models if you're using these polymicrobial systems. You can't simply add equimolar amounts of organisms together, throw them in the tube and expect the biofilm to grow. In order to have them be reliable and reproducible, you need to do that. And they can be used to both screen the actives, evaluate inflammatory responses, and we've also used it to, you can exchange these organisms for mutants, for example, mutants of P. gingivalis or streps, to see how the biofilm dysbioses or changes as response to maybe reductions in adhesions and so on and so forth. So it's a really useful and malleable system to ask questions. So moving on a gear, then what about yeast? So obviously I started off talking about Candida. I think Candida is my organism of choice. So I've got a mission in my mind to show that Candida is important in the oral cavity. Fortunately, a study that just came out of Bangalore has helped me there. So I've had an idea for a while that Candida's, you can find Candida in subgingivally. You find Candida associated with periodontal disease, and there's this new systematic review out which suggests there's a positive association between Candida species and periodontal disease. I'll leave it at that. I think Candida is there. It's important, and I'll sort of go through some slides now to explain why it's important. I don't suggest that it's maybe a key organism that's driving periodontal disease, but I think its physical presence within that environment has a role. So Candida, as Alex said, do support bacteria. We know, and I think again Alex alluded to the fact that you find a lot less of it, and that's not surprising. So yeast cells are approximately about 75 to 100 times the biovolume of bacteria. So this is a hyphae. You can see that all these little bacteria, this is Staph aureus, attaching to Candida hyphae. But in terms of the yeast part of that, then it's around about 100 times biovolume. So when you see 2 logs or 3 logs reduction in Candida compared to bacteria, that's normal. I mean, that would take up the same space as those bacteria. There's just less of it. But they occupy this high physical volume. They provide these scaffolds, which we term microfilms, that we know that there is an exchange of secreted metabolic products, which can either be antagonistic or synergistic. And one of the important things, and Bastien Kroon from ACTA in Amsterdam has shown, and we can sort of replicate these experiments, is that if you put P. gingivalis, which absolutely won't grow out with the anaerobic cabinet, if you put it along with Candida albicans, it'll grow, suggesting that Candida albicans quickly drives down the oxygen tension, allowing anaerobes to grow within. So Candida has a supporting role. And indeed, we've done some studies along with Richard Lamont and our own labs showing that there's an absolute association between things like Candida and P. gingivalis. We know that P. gingivalis has these internalins which help attach to Candida, and similarly Candida has ALS3. So if you knock out ALS3 in Candida, then P. gingivalis doesn't stick so much. And similarly with Candida and Staph aureus, we know that you can see these intimate associations. And again, if you knock out ALS3 in Candida, then Staph can't. So we know that Candida is really important, allowing different bacteria to attach. So Chris Delaney, who's just completed his PhD and is a postdoc with me, put together this paper a couple of years ago. It's not on PubMed, but you can find it. It's easily downloadable. But it kind of just goes through sequentially the different oral sites where Candida has been found, and there's lots of information on these co-associations, whether it's Enterococcus faecalis, whether it's Lactobacillus, whether it's Staph aureus, whether it's P. gingivalis. There is a lot of information showing the specific molecular interactions, and I think what we're seeing now is that a lot of people in the fungal field are starting to show an interest in bacteria, and a few people in the bacterial field are starting to show an interest in fungi. It was quite notable, and maybe Alex will have a right to reply after my talk, but none of the microbiome studies that he mentioned touched on the fact that Candida was around, and I think there is a lack of microbiome research with respect to the oral cavity, and I think that could be quite illuminating. But nonetheless, so there is a real mechanistic importance of how Candida interact with bacteria. So understanding the physical interactions is probably important in terms of understanding how these organisms drive different sort of, or how different inflammatory processes happen, because maybe Candida facilitates the bacteria to be there in a way that we don't consider. So I kind of alluded to the fact that everyone thinks about P. gingivalis as the keystone organism. So I've actually put in a little diagram here just to illustrate what keystone actually means. So the idea that really that sort of in old Roman architecture, if you put the keystone in, that supports the rest of the structure. And if you remove that keystone, the whole structure will simply fall apart, similar to, I think, the idea of Fusobacterium nucleatum. So we've shown with Tracy, so Tracy's just about to get her PhD viva, but we've got a hard tissue model, which basically is a caries model, which has about 5 different species, including Candida, and our soft tissue model, which is essentially our 10 species model plus Candida albicans. And if you look at the number of bacteria in these systems, if you remove Candida, you see at least a log reduction. So in the caries model, you see over a log reduction of bacteria, and similar with the soft tissue, the periotype model, we see a reduction.
Professor Ramaj, can I disturb you for a minute? Can you please switch off your camera, because the screen is appearing very blurred?
Oh, sorry. I hadn't realised it was on. I can't find my camera.
Video, video, sir. Camera, where?
Oh, sick.
It's better.
Is that it?
Yes, it's better now. Sure.
Okay, sorry.
Thank you.
Yeah, so what we're able to show is that we get this reduction in bacteria if Candida is not there, and you can see quite clearly that the composition of the biofilm changes when you add Candida into the system. So here, when you've not got Candida, then the anus elandi takes over, whereas you put it back in, then F. nuc actually starts to become more important. And similarly, in the more complex system, the soft tissue model, as we call it, then we see a reduction in A, and we start to see more proportions of even P. gingivalis and so on. So you do get these changes in dynamics. So you can then use those systems to then look at the effects of treatment and virulence. So, for example, we use this live dead PCR approach, so we can look at viability in the biofilm system. So we use Propidium monoazide. So if you have live cells, then the Propidium monoazide cannot penetrate inside the cell and intercalate with the DNA, whereas if you have dead cells, then the PMMA interacts with the DNA. So when you do a PCR reaction, you cannot amplify the dead DNA. So all you're doing is amplifying viable DNA. Okay, so again, this paper by Jason will come out shortly, but it's a really useful way in which you can measure viable cells using a molecular method. And using that methodology, we're able to show that if you add Candida to the system, whether it's treated with chlorhexidine, EDTA, potassium iodide, miconazole, then they're much more resilient or tolerant to antimicrobial activity, both in our hard tissue model and our soft tissue model. And I think we looked at pH as well. So, I think we know that Candida can actually drive down the pH, but it can also buffer pH as well. And in both our models, we were able to show that Candida actually takes our models back toward a more neutral pH. The reduced lactate, in the hard tissue model, it reduced superoxide dismutases, whereas in the soft tissue model, it increased it. And then we're seeing increases in protease. So there is changes in virulence attributes as well as treatment. And what we know from our own microbiome studies in a denture context, this is the work that Christopher did, and this is the key thing. So when we look at our clinical study in a whole range of patients who've either done excellent oral care, good oral care, or poor oral hygiene, irrespective of how you manage your mouth, the Candida levels seem to be maintained. So Candida is clearly important, and I think it's very difficult to perturb Candida. So it seems to be a dominant. It's not numerically dominant, but it's resilient enough. And I think back to Alex's analogy in terms of how the original oral systems operate is you need these pioneers, and I think Candida is part of one of those pioneers organisms, and it's very difficult to remove it. So once your system's there and intact, then it will just regrow and regrow. So we have done some microbiome studies, and so this is Lindsay, who did some studies with ACTA, and we're interested on how our denture microbiome compared to our mucosal microbiome, compared to our gingival microbiome, if you like. So you can see quite clearly that what's on the denture is quite different to what's in your intact dentition. So that's kind of important. And we can show there's more diversity in your dental plaque than there is on a denture. And we can show that using these PCA plots. I suppose what was more interesting to me is, because of my focus on Candida, is that as you increase the Candida burden, you can see this yellow line sort of going down a bit, and we can do some sort of multivariate analysis, and we can show that there's an association with Candida and Lactobacillus, and then other organisms such as Streptococci go the other way, or some of the anaerobes go the other way. So clearly, as Candida goes up, suggesting dysbiosis, then that changes the types of organisms that we see in these patient groups. But I think one of the key things we showed from this study was that obviously denture plaque and dental plaque are quite different. But the reason why we've done these studies is we can then use what we find from these microbiome studies to then redefine some of our models. And indeed, we've done that to a certain extent. So Leanne, who was with us from 2010 to 2016, she was the one that sort of developed this sort of live dead PCR assay. And we've done a whole range of treatments to assess. You know, it's strange, it's arguable that there's no real clear guidance in how we should manage dentures, and you know, some people not be very excited about that, but clear clinical guidance is important. So you need a denture cleanser, you know, so there's do you use a denture cleanser then brush, or do you brush then use a denture cleanser? And what we showed that it does have an effect by brushing and then using a denture cleanser. You can certainly reduce by almost 3 logs, the amount of organisms are there, but also change the proportions of organisms. So depending how you manage the denture, and you can change the numbers and the quantity. But that was a kind of a proof of concept type study, and that led to another GSK study where we wanted to look at the impact of denture cleaning, not only in the lab, but use that as a segue to do a clinical trial. So without going into huge detail, really, then we're able to show using this PCA that this is untreated. This is our intermittent, and this is our denture treatment. So basically, we can see this is based on microbiological parameters. We can see quite a difference in terms of how we manage them, and that's the idea that you treat with a denture cleanser daily, rather than what most people do is take a denture cleanser on day one, then they might brush it through the week, and then maybe at the weekend they use the denture cleanser again. So you can see quite a distinct microbial pattern depending on how you manage the patient. So when you take it into real patients, so we took it into about 17 or 18 patients and did a double examiner blind randomized double blind crossover, where the patients either had a sequential denture cleansing for the first week and then switched to the intermittent, or vice versa. And then we looked at some microbial and clinical parameters. So we took measurements at these different time points. And whether you've got aerobes or anaerobes, what we showed was the blues, the bit that we're interested in, the daily use of denture cleansers, as is predictable, showed a reduction in microbial burden. We're able to show changes in, you know, this is somewhat artificial because we're looking at the same organisms from the in vitro. We did try to do microbiome analysis, but the amount of sample to do it was simply too little, and we couldn't get anything good back. But nonetheless, you see a slight change, well, certainly a different pattern in terms of the microbial organisms present. And from a clinical point of view, then we see a reduction in plaque scores, and the sort of different patient groups. So ultimately, the models which we conducted in the laboratory were able to inform the clinical trial design, which was then able to show and demonstrate unequivocally that daily denture cleansing is more effective than intermittent. And I know it's a logical outcome, but there was simply no evidence to show that in the literature to this point. So just before I finish up, so Candida and other yeast are certainly important components of the oral biofilm. So hopefully that answers the last question of Alex's talk. Candida does clearly impact antimicrobial activity and oral hygiene practices. And I think it's really important to understand the oral microbiome and oral mycobiome, because I think both together will give us a better, more deeper understanding of how these pathogenic processes drive oral disease. So just for the last minute or two, I want to just touch on where we're going, so I won't take up too much time. So this is Candida albicans, P. gingivalis, and Pseudomonas aeruginosa. So we're able to have these probes to specifically look at these organisms. And I think that's a point that Alex made as well, is that microbiome research is great, but it doesn't give you the whole picture. You need to see what these organisms are doing together to try and get an idea of what's going on. But what we're trying to do as a group at the moment is we're trying to create a library of all microbiome studies that have been conducted in the oral cavity. So we are funded by GSK, and we are actively data mining every microbiome study that's been done since it started. So we're going through literature screening. So we're screening all the literature, we're finding every study. We are quality controlling all of these studies, and we're trying to then bring all the data into a single site. The difficulty is that people aren't very good at storing data. So they publish microbiome studies, but they don't provide the data, although they're meant to. They state in their methodology that it's there. When you go and try and find the data, it's not there. So there is issues with that. But the idea is to be able to take these data together from caries, perio, endo, oral cancer, for example, and then create a database of all the studies, which has then become queryable, where you can then plug in your own microbiome data set and see how that fits with all the control data from, for example, healthy individuals or those with gingivitis or perio. And the idea really is that, I say it can be queryable. So that's kind of what we're doing at the moment. One of the issues I have, I suppose, is sampling in the oral cavity. And I think the data that you get out at the end is only as good as the data you put in. So unless these studies have very definitive methodology statements, the metadata suggests where the sample was taken from, how the sample was taken, how it was processed, so on, that needs to be standardized and uniform. Otherwise, so what you've got, for example, sorry, I'm just going to batter on here. This is my tooth surface, if you like. And if you think about taking a swab, clearly here, this is a site in Brazil. This is the favelas, and over there on the other side here, you've got a more affluent area. But if you were to take a swab across this particular biofilm on the tooth surface, then you're going to get a bit of both. And that does not represent what's there. But when you do the analysis, you're going to get, these will be representing different bacterial species. These will represent different bacterial species, and it will suggest that there's some sort of interaction. So we've got to be really careful about how we take samples. And as we've become better at the analytical process, we need to certainly have a think about and go back and really have a clear idea what the sampling processes are and processing are, because if not, then the microbiome studies that we're developing are going to be kind of useless. And I think, I think we need to combine imaging with microbiome sampling. I think we need to have that functional outputs. We need to have the physical to see what these organisms are doing in order to have a better understanding of what they're doing within our model systems. So going forward, what we're really going to do is we're now in a position we can take these multi-species consortia direct from patients' mouths, and rather than taking these models which we've so arduously put together, we can then just use those and use things like nanopore and other Illumina sequencing platforms and have our measurable outputs, because it's much more inexpensive now compared to what we do here, where we're using PCR. It's much more labor intensive and arguably more expensive. So there's different ways in which we can now manage or interrogate our biofilm data. So to conclude, hopefully I've shown you biofilm models are useful tools to undertake controlled experiments. Next generation sequencing has definitely improved our way to inform and refine our models. And I think it's really going to give a useful platform to conduct more meaningful clinical sample type experiments. So even from a clinical perspective in the lab, so preclinical, but with more applicable biofilm consortia. And I think beyond anything, I think hopefully I've shown that working with a company like GSK has allowed you allowed us as a group to develop relationships with industry, and it can be a really positive experience. And certainly it's kept our lab going for the last, how many years that's, 13, 14 years. So thank you to my sponsors. And here's a number of the people who have been doing the work for us over the years. So some of them popped up in the presentation. Some of them are, Endo and Candida and all other manners of things. So thank you very much for your attention, and hopefully I haven't gone too much over time. Thank you.
Thank you, Professor Ramesh, for this enlightening lecture. Here we have some questions from the audience. Now, there's a question, why not study oral microcosm rather than studying multi-species biofilm? Is it more realistic?
Well, it's not more realistic. I think the reality of it is, and I think hopefully I touched it at the end, back in 2010 or so, then what would have been required to use real microcosms to analyze it in detail would have been very expensive and laborious. So what we've taken is a halfway house where we've taken key organisms in a controlled manner, because I think ultimately any scientist will tell you that unless you've got controls and reproducibility, then your experimental outcomes are diminished. So yes, absolutely take the point. Microcosms are probably better, and I think that's now where we're in a position we can do that. But until then, you need to work with something, and that's kind of what we've done.
Okay, thank you. I'm just curious to know, what are the substrates that you use for growing these biofilms in your laboratory?
So we tend to use a range of substrates. For more high throughput, we use 96 well plates, which are polystyrene, and they've got no relevance to the oral cavity, but again, they use that reproducibility. We regularly use hydroxyapatite or hydroxyapatite discs, and for a lot of the endo work, we use dentine discs. And for a lot of the denture work, we use PMMA. So we use substrates relevant to what we're doing, and then obviously for the inflammatory work, we've tried to put together organisms on hydroxyapatite next to epithelial cells. So we just have to try and be as creative as possible.
Other than Candida albicans, have you looked at any other fungi in the oral biofilms?
Not in the oral biofilms. So I think
biofilms, have you used any other organism other than the Candida albicans in terms of fungi?
We do lots of other work in fungi, but from things like Aspergillus and other yeasts. But to be honest, the only organisms that are really important in the oral cavity are yeast. There are some studies in microbiome that come up with a whole bunch of other organisms, but I would argue that those, because you're breathing in spores and so on from filamentous fungi, they just happen to be passing through your mouth, and they're probably part of what you're eating. So they're not resident endogenous microflora. So things like Saccharomyces cerevisiae and other Candida species, absolutely. But beyond that, you're just got then they're not participants in the oral microbiome.
Now, how do you harvest these or the biofilm that is grown in vitro for further analysis?
So we tend to use a sonic bath. So we would tend to remove the substrate and then place it into a receptacle which goes into a sonic bath. So the sonic waves then basically remove this from surface. So that was learning from my PhD when we did that with prosthetic joints directly from patients. But if you put a denture, even if you have a denture patient, if you had a sonic bath and put the denture within an enclosed plastic bag with water or saline, it's quite remarkable the amount of organisms that come flying off that surface. So it's a very useful way to harvest them. Some people try and scrape them, but you don't get all the organisms that way.
Instead of growing biofilms in vitro, is it possible to use any animal models to grow these biofilms and then use it for further understanding of the host microbial interactions, or even to know?
Yeah. I mean, you can, and I think I'll leave that. I'm pretty sure Professor Culshaw will probably come to that at some point. But there are various models that are appropriate. But yeah, I'm gonna leave that to Shauna to talk about.
Sure. Thank you, sir. I now request Dr. Faizuddin sir, Chairman, Dental Scientific Committee, Silver Jubilee Celebration, Rajiv Gandhi University of Health Sciences, to hand over a certificate of appreciation to Professor Ramaj.
Indeed, it is a great pleasure, Professor Ramaj, to present this certificate to you on behalf of Rajiv Gandhi University of Health Sciences, on behalf of Bangalore Academy of Periodontology, and on behalf of VS Dental College. We are very grateful to you for accepting our invitation and having enlightened us on the various models of biofilm. Thank you.
Thank you very much. It's been a great pleasure to talk to you, and if anyone wants to talk further about the work we do, then I'm happy to communicate by email or Zoom. Okay, so thank you very much.
We will continue this relationship. I think we will have a long-lasting relationship and we'll keep exchanging our views. Thank you.
Thank you.
Thank you so much. Thank you, sir. Now I invite Dr. Aruna, Professor, Department of Periodontics, to moderate the next two lectures. Thank you, Dr. Sushma. Greetings from VS Dental College and Hospital. It's my pleasure to introduce our next speaker of today, Mr. William Johnston. He has a first-class honors in biomedical science and was awarded the President's Prize for the highest academic achievement. Currently, he is a research scholar at the University of Glasgow, focusing on how the immune system and subgingival plaque microbiome change following periodontal therapy. He has 5 publications and a grant to his credit for his research work. During his PhD, today he is presenting his work on analyzing the immune response and plaque microbiome after periodontal therapy. Over to you, sir.
Thank you very much for the lovely introduction. I'll just check everyone can see my screen okay, and is that okay? Everyone hear me and see? Okay, so thank you for the lovely introduction, and thank you for inviting me to speak today. My name's obviously William Johnston, and I'm a PhD student in the University of Glasgow at the Dental Hospital, where I work alongside Gordon and Shauna, and we also collaborate with Alex quite a lot as well on some of the microbiome stuff. And we've seen 2 really great talks this morning, looking at the role of the oral microbiome and what influence this has on various other systemic diseases. And I think my presentation is going to be slightly different because I've obviously not covered as much work as some of the other professors that we've heard today. And I'm really only going to focus on the work that I've been able to conduct throughout my PhD project. And specifically, I'm going to talk a bit about a clinical trial that we ran where we were looking to analyse how the immune response, both locally and systemically, and the subgingival plaque microbiome change following periodontal treatment, and specifically following non-surgical periodontal therapy. And throughout the presentation today, I'm really going to highlight how we conducted the trial, how we processed the patient samples, which, as we've heard Gordon talking about, is a really important aspect of clinical research, and also highlight some of the data that we've been able to generate and what this means in the wider context. So this is just a very basic overview of what I'm going to be talking about. So I'll start by going through a bit of background into our clinical trial, talking about why we did it and what we were hoping to find out. I'll then go through our methodology section, focusing on our study design and sample processing techniques. And then I'll show some of the data that we've been able to generate. And I'm going to focus on serum, saliva, and subgingival plaque samples, of course. And then I'll finish up by giving a brief summary of what we found, what this means, and also highlight some directions that we've been looking in terms of future work to stem out from this project. And you can probably tell already that the presentation isn't going to be solely focused on the subgingival plaque microbiome, although I will cover it. And hopefully the stuff that I discussed beforehand in the serum and saliva will lead quite nicely into why we've done this analysis, and also highlight the very important point, as we've heard already this morning, that quite often the impact of bacteria and fungi in the oral cavity, and specifically in the subgingival plaque, the impact that these organisms can have can often extend well beyond the oral cavity and have quite drastic systemic implications. And hopefully I'll highlight some of these throughout my presentation this morning. So the study that I'm going to be talking through today is called the IRAPT trial, which stands for the immune response after periodontal treatment. And we published some of the outcomes from this study in the middle of last year in the Journal of Clinical Periodontology, which is really going to be the sort of basis of this presentation. So if after this talk anyone wants to see some more of the data, or perhaps go through some of our methods in a bit more detail, then they should all be present in this manuscript here, and I can show this again at the end if anyone's interested. So the IRAP trial that we conducted throughout my PhD was a randomized controlled trial that was based out of the dental hospital in Glasgow within our clinical research facility that we have access to, that I've shown in this lovely image here. And they also have their own Twitter feed if anyone's interested in keeping up to date with some of the other work that goes on in our lab. Now, throughout the current study, we were aiming to compare the immediate inflammatory and microbial alterations within periodontitis patients following full mouth debridement conducted using either hand or ultrasonic scalers. So we were wanting to compare the differences when treatment is conducted using these two different types of instruments. And I've placed quite a lot of emphasis here on the word immediate, because we were obviously looking at these variables from the immune perspective and also the microbial perspective at a very early stage following treatment, which is quite unusual, and it's in contrast to what you'll find quite a lot of studies looking at, which is obviously the long-term response from a microbial and inflammatory perspective. So why were we interested in this immediate response? Why did we want to look at these factors so early following non-surgical treatment? Well, as I've highlighted in these three graphs here, in the last sort of 10 years or so, it's been noticed that one day following full mouth debridement, so intensive non-surgical treatment where the whole mouth is treated within a single day, you get quite a large spike in systemic inflammatory markers. So as I've highlighted in this graph here, things like serum C-reactive protein, which is a non-specific marker of systemic inflammation, as well as interleukin-6 and tumor necrosis factor alpha, all show this rapid spike following full mouth debridement. And what's quite interesting is that this study also found that this increase isn't observed following quadrant debridement, where treatment is performed obviously in quadrants and is often spread across several days or several weeks. Now, the important thing here is that this spike that we see following intensive full mouth debridement is thought to be due to two quite different factors. The first one is local trauma. So it's hypothesized that during these intensive treatment sessions, cells within the gingival tissue can become damaged and begin to release pro-inflammatory cytokines, which then circulate throughout the rest of the body. And the other one is bacteremia, which we heard Alex talking a bit about this morning. So it's believed that bacteria from the subgingival plaque, or perhaps other sites in the mouth, can actually enter the bloodstream during these intensive treatment sessions and cause quite a large increase in the day one levels of all these inflammatory markers. Now, what is also important to note, I think, is whilst we do see a rapid increase in all three of these inflammatory markers, all of them return back to baseline levels by day 7, so after 1 week, which would suggest that this is only a very transient response that we're observing within these patients. And this spike in systemic inflammation following full mouth debridement has been shown in loads of independent studies. So I've shown 3 different studies up the top here that have all looked at the level of serum C-reactive protein 1 day following full mouth debridement, or intensive treatment as it's quite often referred to. And you can see for each study, you get this rapid spike in the level of C-reactive protein. Now, probably the most relevant finding in relation to the study that we conducted throughout my PhD project is the one that I've highlighted in this red box here, because this study in 2015 found that the degree or the extent of this systemic inflammatory response positively correlates with treatment time. So longer treatments appear to be associated with a larger systemic inflammatory response in comparison with shorter treatments. And again, it's believed that this ties in to things like the local dissemination of cytokines and also bacteremia that's induced via the treatment sessions. And obviously, this response that we see following full mouth debridement is quite unusual and it's quite interesting. But the real question is, why do we care about it? Why is it clinically relevant? And to be honest, there's been a lot of debate about the clinical significance of this systemic inflammatory response, because on one hand, as we've seen throughout all of these previous studies, all of the inflammatory markers return back to baseline levels by day 7. So it's clearly a very short-lived and transient response. But on the other hand, you've got the argument that in patients with underlying health conditions, for example, things like cardiovascular disease, CVD, this rapid increase in systemic inflammation may potentially exacerbate some of these pre-existing issues. And in agreement with the latter, the European Federation of Periodontology and the American Academy of Periodontology released a joint consensus statement last year, again in Journal of Clinical Perio, which advises against the use of full mouth debridement in patients with any level of cardiovascular disease, so even very, very mild forms. And so it appears like these concerns about exacerbating pre-existing issues may actually carry some merit because it's now been implemented into clinical practice. So how did all of this previous literature filter in and relate to the IRAP study? Well, as we've seen, there's been a lot of studies looking at this inflammatory response and comparing comparing full mouth debridement against quadrant therapy or against surgical therapy, for example. But what all of these studies have in common is that they all use a blended approach when it comes to instrumentation in terms of combining the use of hand and ultrasonic scalers when they're performing the full mouth debridement. So from our perspective, throughout the current study, we were wanting to investigate whether this systemic response would differ, specifically at day 1, if the treatment was conducted using either only hand instruments or only ultrasonic instruments. And the sort of thought process behind this was that some previous literature has suggested differences in treatment time when you perform the treatment using these two different instruments, which, as we've seen, could potentially relate to the systemic inflammatory response. And also hand and ultrasonics have quite different mechanisms of action, which could potentially relate to things like the dissemination of cytokines or perhaps the amount of bacteremia that they're inducing. Now, along with looking at the systemic inflammatory response, we also included some secondary outcomes where we looked at the local inflammatory response within the saliva, and of course, looking at the microbial response within the subgingival plaque and the microbiome, which I'll talk about as the talk progresses. Now, on this slide, I've just highlighted a really basic overview of our trial design, which, as I mentioned at the start of the presentation, was a randomized controlled trial. And throughout this trial, we recruited 37 periodontitis patients. And all of these patients received full mouth debridement, except 19 had treatment using only hand scalers and 18 had their treatment using only ultrasonics. And then following treatment, patients returned at day 1, day 7, and day 90, where we collected all of our different samples and measured some clinical parameters. And these time points were selected so that we could capture this initial spike at day 1, but also see how this inflammatory response differs and recovers over a longer period. Now, one thing that I wanted to highlight is that based on our sample size here of 37 patients, we were powered to detect a difference in C-reactive protein of 3.5 milligrams per litre at day 1 between our two groups here. And I think this value is quite important to bear in mind when I go on and show some of our data. So what did we actually find from this trial? And moving on to the results section, I've essentially split this up into the different sample types that we collected. So we've got serum, saliva, and subgingival plaque, so that I can initially show you how we processed each of these sample types, but also show the corresponding data that we've been able to generate from each one. But I initially wanted to start off very, very briefly and show some data that we've generated from a clinical perspective, which I'm sure most people will be quite familiar with. And I wanted to highlight this initially because I think it ties in to some of the decisions and some of the hypotheses that we've made as the trial has progressed. Now, initially, the first thing that we wanted to look at from a clinical perspective was, of course, how long treatment actually took when it was conducted using hand or ultrasonics. And I've shown the total treatment time in minutes in this graph on the right-hand side, with the ultrasonics shown in blue and the hand-scaled patients shown in red. And you can see, looking at this graph here, that throughout our study, we found that ultrasonics were significantly quicker than hand scalers. And this analysis was adjusted for both the total number of teeth that they were scaling and the disease severity of patients at baseline. So even when we factored these things in to our statistical analysis, ultrasonics were still significantly quicker. And we found that the difference between them on average was about 25 minutes. So the ultrasonic time at ultrasonic scalers took around 7 to 8 minutes, whereas hand scalers took around 96 minutes to perform this full mouth debridement. We then went on to look at changes in actual clinical parameters. And obviously throughout our periodontal examination, we measure quite a lot of different parameters. But for simplicity, I've just shown one on the right-hand side, which is the percentage of sites within patient's mouth, percentage of pockets that were greater than or equal to 5 millimeters. So essentially the percentage of deep pockets. And I've shown the ultrasonic patients again in blue and the hand-scaled patients again in red at baseline and day 90. And again, you can tell looking at this graph that both hand and ultrasonic scalers significantly reduced the percentage of deep pockets following treatment, and there was no difference between instruments in this clinical improvement. I think this is really important to highlight, and this was the same for all of the periodontal parameters that we looked at, things like clinical attachment level, full mouth pocket depth, leading on probing, plaque scores, all showed a very, very similar trend to what we're observing here. So although ultrasonics are quicker, they still perform equally as well from a clinical perspective, which is exactly what we're expecting to find. Now, having seen a significant difference in treatment time, we then went on to look in the serum and begin to look at some of our primary outcomes from the study, which was serum C-reactive protein. And I've initially highlighted on this slide how we actually processed the serum samples, how we got from our patients sitting in dental chairs to clinical samples that we can then go on and use to analyze things like C-reactive protein and different pro-inflammatory cytokines. And to be honest, for processing and collecting serum samples, we used a really, really standardized method, which is fairly consistent across a lot of different studies that you'll read, probably the majority of different studies. And as we heard Professor Ramage talking about earlier, having this consistent standardized collection and processing technique is really, really essential for clinical research, because it allows you to compare the results of your study against other studies that have been conducted previously. So for serum samples, we used this standardized method. And what we did was we collected samples using venipuncture, and we collected them into these specialized tubes which contain clot activators. And throughout the current study, we used silica particles, but there's loads of alternatives which essentially do the same thing. You want the blood to clot. So after we collected these samples, we left the tubes at room temperature for about 25 minutes in order to facilitate this coagulation process. And then we centrifuged them down in order to separate our red blood cells, shown down the bottom here in red, from our serum samples, shown in yellow. And obviously these serum samples are this is the sample type that contains C-reactive protein, interleukin-6, TNF-alpha, for example. What we then done prior to storing it was we then aliquoted this into lower working volumes, things like 100, 200, maybe 500 microliters, so that when it came to the analysis stage, we didn't have to continually freeze thaw the same big sample type, because we know that this influences the levels of different inflammatory markers that we might be interested in. So again, this aliquoting step is really, really important for the quality of data that you'll get back from your samples. And then once we had these aliquots, we could, of course, then store them. Once we had the samples, we could then go on and analyze our outcomes. And I've shown on this slide the level of serum C-reactive protein, which was the primary outcome for the study. It's what the whole study was powered to detect. And you can see in this graph here, looking at the level of C-reactive protein, again, the ultrasonic patients are shown in blue, and the hand-scaled patients are shown in red. And I've plotted this at every single time point, baseline, day 1, day 7, day 90. And looking at this graph here, you can see that both groups follow a very, very similar trend. So we get this increase in C-reactive protein at day 1, following hand and ultrasonic scaling. And then we see a gradual recovery at day 7 and day 90. So very similar trend to what has been observed in these previous studies. We then went on to look at whether the level differed significantly between our two groups, focusing specifically at day 1. And we did this using a general linear model. And what we found, you can tell already looking in the graph here, is that there was absolutely no difference in CRP between our two groups. And this was consistent at day 7 and day 90. And this was true even if we adjusted our statistical analysis for different covariates, things like patient's age, smoking status, BMI, gender. Even when we filtered these things into our analysis, it didn't change the fact that there was no difference between hand or ultrasonic scaling, specifically at day 1. And this was true for interleukin-6 as well and tumor necrosis factor alpha. So very similar trend. We saw an increase at day 1, but there was no difference between our two groups, which you can imagine, given that this was what the whole study was powered to detect, was relatively disappointing from our point of view. Now, whilst we didn't observe a significant difference between our two groups, all of these patients were receiving full mouth debridement. And we did observe this systemic inflammatory response that we were looking for. So if we look on this graph on the right-hand side, I've shown the level of C-reactive protein across all patients, all 37 patients, at each of our time points. And you can see a really clear trend here. Significant increase at day 1, followed by a gradual recovery at day 7 and day 90. Now, what was very, very interesting and quite surprising from our perspective, looking at these data from the current study, is that if we look at the error bars here on this day 1 bar, there seemed to be quite a large inter-patient variation. And what I mean by this is that some patients showed a really, really large increase in CRP at day 1, whereas other patients actually showed no or very, very little change. And given that, having seen the results from the previous slide, this clearly was not happening due to what instrument these patients were being treated with. So we then started to hypothesize different mechanisms that might be contributing to this inflammatory response and whether we could use these mechanisms to understand why there was such a large variation within our patient cohort. And one thing that we went on to look at was antibacterial antibody titers within the serum samples. And the sort of thought process behind this was that, as we've seen, one theory as to how the systemic inflammatory response occurs is through bacteremia induced during these intensive treatment sessions. And so we hypothesized that if bacteria were entering the bloodstream during the treatment sessions, then we might be able to see an increased antibody titer directed against them, specifically at day 1 or day 7, for example. So we chose 4 common periodontal pathogens that we've heard a lot about already: Porphyromonas gingivalis, Prevotella intermedia, Aggregatibacter actinomyces temcomitans, and Fusobacterium nucleatum. And we looked in patients' serum samples for IgG antibodies directed against these 4 species. Now, I've plotted the data here in these graphs, and the antibody titers per patient are shown as ELISA units. And again, similar to the previous graphs, the ultrasonic patients are shown in blue, and the hand-scaled patients are shown in red. And what was very, very surprising is that when we looked at the level of these antibodies, we actually observed no difference, either looking across time points, looking longitudinally within each group, comparing baseline day 1, day 7, for example, or between groups at any of these time points. And this was obviously really surprising, given that we were identifying this significant increase in C-reactive protein, and we would hypothesize that this could be due to things like differences in the amount of bacteremia that these patients are undergoing. Now, I guess a key caveat to mention with this analysis is that clearly we've only looked for antibodies directed against 4 lab species of bacteria. Now, as we've seen earlier on, there's about 100 or 200 different species actually colonize the subgingival plaque. And so it's possible that this response could be occurring. We could be seeing an increased antibody titer directed against some species, but we could just be missing it because we're only looking at a relatively small number of organisms. But I think what's important to highlight is that these 4 organisms are fairly commonly isolated from the subgingival plaque, and this was the case throughout the current cohort when we go on and look at the microbiome analysis later on. And certainly when we looked at antibodies against these 4 species, there was absolutely no difference. Now, having seen this data from the serum, looking at the systemic response, looking at C-reactive protein, different antibody titers, it probably won't come as too much of a shock that we then wanted to take a step back and move away from the serum and start to look at a bit more of a local response. And again, the thought process behind this analysis was that another theory as to how the systemic response occurs is through local trauma and cytokine release within the gingival tissue. So we wanted to see whether we could capture this local response in saliva samples and start to see whether this may explain the variations that we observed within our cohort. Now, initially, to collect and process saliva samples, I've simply highlighted the method on this slide that we used throughout the current study. Now, in contrast to the serum, where we had this really consistent and standardized collection and processing method, in saliva, for saliva samples, there's a lot of different ways that this can be done. And I've highlighted this saliva collection handbook by a company called Salimetrics down the bottom here, because it really highlights the advantages and disadvantages of each one. So throughout the current study, we collected whole unstimulated saliva samples using the passive drool method. So we essentially asked patients to drool continuously for a set amount of time into these 50 milliliter plastic tubes. Now, the alternatives to this are things like salivary swabs, or using stimulated saliva with paraffin wax, for example, which each of these different collection methods has their own advantages and disadvantages. From our perspective in the current study, using whole unstimulated saliva is by far the most popular. And what this allows us to do is compare our results to those conducted previously with the similar collection method. Once we collected the samples, we then aliquoted them into these smaller volumes and then centrifuged them down at a fairly high speed, 10,000 RPM, in order to obtain pelleted saliva. And this step is really, really important. I think it's really useful to understand the differences between the sort of downstream analysis that can be done on saliva, because we then split our pelleted saliva into 2 independent sample types. And the analysis that you go on and perform from each one is very, very different. So the first one up the top here is what's called the salivary pellet. So essentially everything that has been collected at the bottom of the tube here. And this is quite often used to look for things like the bacterial content or the level of different microorganisms, different fungi within saliva samples. You'll see a lot of recent studies looking at the salivary microbiome, and this is quite often the sample type that they will use to conduct this analysis. Now, from our perspective in the current study, from a salivary perspective, we were mainly interested in the inflammatory response. And so what we did was we took the supernatant from this tube here, which is known as the clarified saliva. And this contains all of your sort of immunological markers, things like pro-inflammatory cytokines, levels of different antibodies, for example. And that's what we then went on to analyze. Now, in contrast to the serum samples, where things like C-reactive protein, interleukin-6, tumor necrosis factor alpha, are really quite well-defined markers of this systemic response that we see to full-mouth debridement. In saliva samples, we were really going into this analysis completely blind, because no study has ever looked at the local inflammatory response following full-mouth debridement. And so we didn't know which markers would be present in our samples, and if they were present, whether we'd actually be able to detect them, given all the other stuff that we find in saliva, such as mucins and different bacterial antigens, for example. So initially we took a very, very exploratory approach, and we selected a very small number of patients, 6 from the ultrasonic group and 6 from the hand group. And we ran their baseline and day 1 saliva samples on a Luminex assay that contains 30 chemokines and cytokines. And what these Luminex assays allow you to do, and there's alternatives to this as well, such as things like Olink, which I'm not going to cover in any great detail, but essentially what they allow you to do is look for a really large number of molecules, large number of analytes in a small number of samples, and really get an idea of what is going to be of interest to you throughout your sort of downstream analysis. And I think this highlights probably one of the most important points that I've found throughout my PhD dealing with a clinical trial, is that it's often quite beneficial to use these very exploratory analysis, Luminex, Olink, for example, as a sort of first step, so that you can then identify what's going to be of interest to you and sort of drill down and go on to analyze this in all of the patients. So this is exactly what we did in the current study, and on this graph on the right-hand side, I've shown the fold change at day 1 in all of these different cytokines and chemokines. And you can tell that for the majority of them, as I've shown in these grey bars, there was either no or very, very little change at day 1. So in contrast to things like C-reactive protein in the serum, there didn't appear to be a spike in any of these salivary chemokines or cytokines. Now, as we move over to the right-hand side and we look in these blue bars here, we did identify a small number of analytes, in particular interleukin-1 beta, tumor necrosis factor alpha, interleukin-8 and interleukin-6, where we really started to observe this spike that we were looking for, because all 4 of these markers showed at least a 2-fold increase at day 1. So at least a doubling was observed at day 1 in comparison with baseline. But of course, it's worth remembering that this is only based off of 12 patients. But once we knew what we wanted to look for, we could then drill down and start to look at this in every single patient. And I've plotted the level of these 4 cytokines across all 37 patients in these 4 graphs here. Again, the ultrasonic patients are shown in blue and the hand-scaled patients are shown in red. And what we found initially when we look in figure A here at salivary interleukin-6 is this huge increase at day 1, far more pronounced than what we found in the serum. For example, if we look in the ultrasonic group here, salivary IL-6 increased from about 5 picograms all the way to about 75 picograms at day 1. So a massive, massive increase. Now, what was very surprising, but also really, really interesting, is that when we compared the day 1 level between our 2 groups, it was significantly higher following ultrasonic scaling. When we then went on to look at salivary interleukin-8 in figure B, we saw a very, very similar trend. So this huge increase was observed at day 1. And again, it was significantly higher following ultrasonic scaling. For tumor necrosis factor alpha in figure D, we did observe a similar response. So we got a significant increase following ultrasonic scaling, but not following hand scaling. But it wasn't significantly different when we directly compared our 2 treatment groups, probably because when you look at the error bars here, there was quite a lot of variation, again, in the degree of this response. Finally, for interleukin-1 beta in figure D, we actually didn't observe a spike at all at day 1 within any group, which was obviously in contrast to what we found in the Luminex assay. But we did see a significant reduction in salivary interleukin-1 beta at day 7 and day 90 within both groups. And this ties in to quite a lot of literature that suggests salivary interleukin-1 beta is quite a good biomarker of periodontal inflammation and consistently reduces following a successful course of non-surgical treatment. Now, going back to these first 2 results here for interleukin-6 and interleukin-8, these are really, really interesting because what they suggest is that following ultrasonic scaling, you may get a larger amount of local inflammation. Essentially, it could be suggested that ultrasonics induce more local inflammation and local trauma during the treatment session. But what's important to note here is that clearly, as we've seen in the serum, this doesn't appear to transition systemically. And also, this analysis that we performed on saliva, it's worth remembering, was a purely exploratory secondary outcome for our work. So whilst we've identified these interesting and unusual results, it's important that this is then repeated in a larger study that's actually powered to detect these differences. So the very last sample type that I wanted to talk through today was, of course, the subgingival plaque and some work that we've done looking at the microbiome, which I'm sure will be of interest given the name of the symposium today. And really, from the perspective of the current study, we were wanting to investigate whether the subgingival plaque microbiome differs within periodontitis patients who had been treated using hand or ultrasonic scalers. And again, relating back to the different mechanisms of action of each instrument and hypothesizing that this may play a role and drive differences in how the microbiome initially is impacted by a non-surgical treatment at very early time points, day 1 and day 7, but also how it reforms over a longer time point, thinking about our day 90. So to collect and process subgingival plaque samples, very, very similar to saliva, there's a lot of different ways that this can be done, as we've heard Professor Ramage highlight the problems surrounding this already. And again, I've simply highlighted the method that we used throughout the current study. And to collect subgingival plaque samples, we used curettes. So what we did was we removed the supragingival plaque, and then we essentially scraped the subgingival plaque off of the tooth surface. And we did this from the deepest pocket in the mouth. So we looked at patients' pocket charts at baseline, identified the deepest pocket, which was often a site of about 8 up to 12 millimeters, so really quite deep periodontal pockets. And then we collected plaque from the same pocket at every single subsequent time point. So baseline, day 1, day 7, day 90 samples all came from the same pocket. And this allowed us to view how the subgingival microbiome is impacted by, but also responds to non-surgical treatment within the same subgingival microenvironment. Now, the alternative to using curette samples, as we've seen already, are using things like endodontic paper points. Now, I've highlighted this reference here by Loomer in Periodontology 2000 that highlights the sort of advantages and disadvantages of using curettes versus paper points, because there is microbial differences. When you collect plaque from the same site using curettes and paper points, you can actually observe minute but very prominent differences between each collection method. Now, throughout the current study, we used curette samples, or curettes to sample, because they're able to obtain significantly greater amounts of bacterial DNA. Now, given the time points that we were sampling, for example, day 1 and day 7, for example, we were only expecting very, very low levels of subgingival plaque to actually be present. So we used curettes in order to maximize our chances of being able to retrieve and analyze any subgingival plaque at these early time points. The advantages of using paper points, for example, are you get a little bit greater site specificity. So if you were wanting to compare health versus disease, for example, then it would probably be more beneficial to use endodontic paper points to collect your samples. And I think this highlights a really, really important aspect of clinical research, which is that often it's really beneficial to have an idea of what your downstream analysis is going to be, because the way that you collect, process, and store your clinical samples can actually have a huge influence on the quality of data that you'll receive, or even the ability to perform a certain analysis. So it's something that's worth bearing in mind if anyone's going on to run a trial for themselves. Now, once we collected subgingival plaque, we then put it into Eppendorfs containing sterile PBS. We didn't want any bacteria from tubes or from PBS interfering with our analysis. And then we centrifuged it down in order to obtain a subgingival plaque pellet. Now, I wanted to highlight the point that once you have a subgingival plaque pellet, there's a lot of different downstream analysis pathways that you can go into that aren't limited to looking from a clinical perspective. Although this is what we've done in the current trial, it's worth being aware that there's a lot of different things that you can do once you have this subgingival plaque sample. Now, as we've heard already, you'll see a lot of the earlier studies using things like microbial culture. So they'll take the subgingival plaque and spread it on a lot of different agar plates in order to identify and quantify 1 or 2 or 3 different bacteria, oftentimes things like Porphyromonas gingivalis, for example. More recently, what has become really popular is looking at the microbiome. So you can extract the DNA from this subgingival plaque and perform 16S sequencing, as we heard Dr. Mira talking about in the first presentation, and look at the composition of the microbiome at species or genus level, and look at how this differs between health and disease, or how, in our case, how this responds to a treatment. The other thing that you can do, which we heard Professor Ramage talking about, is you can actually take this subgingival plaque pellet that has been isolated from a periodontitis patient and begin to grow in vitro biofilms for it. And I don't want to cover this too much, I just wanted to mention it. But I think it's worth being aware of and highlighting the point that you can now go on and do these things. And what this allows you to do, these in vitro biofilms, is look at perhaps the impact of novel treatments, or even co-culture these biofilms with immune cells, and look at how an in vitro biofilm that has this huge diversity reflective of a periodontal pocket is actually impacted by these different things. And you can use this in a really, really controlled in vitro setting. And there's a lot of really interesting studies getting released using this sort of similar processing pipeline. Now, without going off topic too much, throughout the remainder of the talk, I purely wanted to focus on the work that we've done in the current study looking at the subgingival plaque microbiome. And initially, when we got this data back, we looked at the composition of the subgingival plaque at the genus level, which I've plotted in this very, very colourful graph here. And I've plotted this at baseline, day 1, day 7, and day 90, with the ultrasonic patients shown on the left-hand side and the hand-scaled patients shown on the right-hand side. And all of these really colourful bars here correspond to the percentage abundance of all of these different bacterial genus that I've highlighted on the right-hand side. And I've tried to arrange them so that the predominantly health-associated genus, things like Streptococcus, Rothia, Actinomyces, Corynebacterium, are highlighted and plotted down the bottom, whereas the predominantly disease-associated genus, things like Porphyromonas, Treponema, Tannerella, Filifactor, are shown up the top. So we should have this nice gradient from most health-associated all the way up to most disease-associated. And of course, the initial question in our mind when we performed this analysis was, does the microbiome differ between hand and ultrasonics? And the short answer is it doesn't, because what we found is that whether we looked at genus or species level at day 1, day 7, or day 90, there was absolutely no difference within the subgingival plaque if treatment was conducted using hand or ultrasonic scalers. Both types of instruments seemed to induce similar immediate impact on the microbiome, and also similar long-term reformation was observed within each group. Now, what we were able to observe throughout the current study, given these quite unique time points that we were sampling, was a really interesting pattern of how the subgingival plaque microbiome responds to non-surgical therapy. And what I mean by this is that if we look at the baseline samples here within both groups, you can see this really diverse and quite evenly split subgingival plaque community, where you've got things like Fusobacterium, Prevotella, Tannerella, Treponema, and Porphyromonas, and these sort of red and brown bars, equating to about 30 or 40% of the total subgingival plaque composition. And these sort of 5 genus are predominantly associated with disease. What we then found at day 1 and day 7 was a progressive increase in the percentage abundance of our health-associated genus down the bottom, and a progressive decrease in these disease-associated genus up the top, to the extent that by day 7, over 50% of the microbiome was now accounted for by things like Streptococcus, Rothia, and Actinomyces, whereas Fusobacterium, Prevotella, Porphyromonas were only present at very, very low levels. And actually, if we look at this day 7 and compare it against previous studies assessing periodontal health, this sort of Streptococcus, Rothia, Actinomyces dominated composition is very similar to what we would see in a periodontally healthy subject with shallow pockets. And this was, so up until day 7, things were looking really, really good. What we then found between day 7 and day 90 was almost like a shift and a reversal back to a composition more similar to what we found at baseline. So if we look at the day 90 composition here, you can see that Porphyromonas, Prevotella, Fusobacterium are starting to re-emerge and begin to increase in abundance, whereas Streptococcus, Rothia, Actinomyces, for example, are starting to decrease in abundance. And overall, the composition at day 90 is becoming much more similar to what it was at baseline. And this is really interesting because it sort of ties in to this notion that it can be really, really difficult to both induce and maintain long-term shifts within the subgingival plaque using mechanical therapy alone. And what we think is happening across these time points is that at day 1 and day 7, immediately following non-surgical treatment, where we're disrupting the biofilm, when we're allowing oxygen to enter into the periodontal pocket, things like Streptococcus and Rothia are simply outcompeting our disease-associated anaerobes up the top, because the environment that we've created during treatment no longer suits the growth of these strict anaerobes. However, as we look at day 7, although the level of these anaerobes is very, very low, they are still present. And by day 90, what we think is happening is that they're simply able to sort of re-establish their niche and begin to repopulate, because although we're significantly reducing the depth of the periodontal pocket, there is still a pocket there, and they can eventually find this anaerobic niche and begin to repopulate. And when we looked at this data, looked at these colourful bars, we thought that this was happening across every single patient. We thought this would be a fairly consistent trend. Now, another way that we've looked at this is what's known as a principal coordinates analysis. And what this allows us to do is plot the composition of the microbiome as a single dot on a two-dimensional curve. And we've already seen very similar graphs presented by Gordon and Alex in the previous presentations. And the way to interpret this is that dots that are closer to one another have a more similar microbiome, and dots that are further away from one another have a more different microbiome. And all of these dots represents the baseline microbiome composition of patients treated with ultrasonics and treated with hand. So if you imagine all of those lovely colourful bars, if you reduce them down for every single patient into a single dot. And what we find when we look at the baseline samples here is that there was a small degree of variation. So not every patient had the same baseline subgingival plaque microbiome, but there was a fairly good degree of consistency between our different patients because they all tended to cluster over on the left-hand side. What we then found at day one, when we look in the yellow samples, is an immediate shift over to the right-hand side. So there was a small degree of overlap. If we look down the bottom left here of the hand scaling, there's some patients clustered very similarly to baseline, which may indicate small areas of the plaque that have been missed during the treatment session. But for the most part, you can see this fairly distinct clustering on the right-hand side, which indicates that not only were the day one samples very different from the baseline samples, but again, there was this degree of consistency that we're observing across all of our different patients, regardless of treatment group. What we then found when we added in the day 7 samples in green is an even more pronounced shift. So even some of the day 1 samples following hand scaling that clustered pretty similarly to baseline, by day 7 had now moved over to the right-hand side and clustered with the sort of day 1 composition. And as we've seen in those lovely colourful graphs, this was clearly indicative of this health-associated compositional shift that we were observing. What was really, really surprising, probably one of the most interesting findings from this trial, or from the microbiome perspective, is that when we added in the day 90 samples in blue, we saw a totally split response. So if we look at the blue dots here within both treatment groups, you can see that some of them clustered very similarly to day 1 and day 7 samples, whereas others started to move back the way and cluster more similarly with the baseline samples. And again, this was happening whether treatment was conducted with ultrasonics or using hand scalers. There was this completely split response. And this is probably clearest when we look at the composition of every single patient on a single graph. Again, all 37 patients. And you can see at baseline in red, fairly consistent clustering on the left-hand side, day 1 in yellow and day 7 in green, fairly consistent clustering on the right-hand side. But for the day 90 samples, we get this totally scattered and random composition that we find. And what this indicates is that following non-surgical treatment at these longer day 90 time points, some patients, the composition moves back the way and starts to become more similar to this disease-associated composition that we find at baseline, whereas other patients, amazingly, are actually able to maintain this health-associated composition that we find at day 1 and day 7. And really moving forward, given these time points that we've sampled, we're hoping to start to drill down and look at this in a lot more detail and start to understand why this response is actually occurring. Now, initially, we originally thought the obvious answer to this would be pocket depth. Does the sites with residual pockets, are these the samples that have this more disease-associated composition? And this isn't the case. All of these samples that we selected were taken from pockets which had a similar baseline pocket depth of about 8 to 12 millimeters, and they all reduced to similar levels following treatment. So this wasn't being driven by pocket depth. And again, I think moving forward with this study, we're hoping to start to really drill down and analyse different factors that we think might be contributing to this response. It'll be interesting to see people's feedback and see what factors they might think would cause such a drastic difference in the long-term microbial response to non-surgical therapy. Now, some other things that we're planning to look at are things like oral hygiene, using our plaque scores that we've got at baseline, day 7 and day 90. Can we use this to identify patients who maybe have poor oral hygiene? Maybe these are the patients where the microbiome starts to shift back the way. Possibly predictive species, thinking from a sort of ecological perspective. can we use our day 1 and day 7 samples and look at the level of certain disease-associated species or groups of species, and use this to identify and investigate whether the level of these organisms at these early time points predict the level that they go to at day 90, or possibly even residual inflammation as well. Looking, obviously, we've got some cytokine analysis in the saliva. We've also got access to gingival clavicular fluid samples. And can we use this sort of biological marker of inflammation to see whether this would shift the composition to reform one way or another? Now, obviously, this is quite a lot of analysis that we didn't originally factor in to our sort of study design and timelines, because we're kind of reacting to the results here as the trial has progressed, but hopefully we'll be able to get this work done and share it very, very soon. So to summarize what we found from the IRAP study, clearly the choice of hand or ultrasonic scalars doesn't appear to influence the systemic inflammatory response one day following full mouth debridement. But as we've seen throughout this trial, there was such a large degree of interpatient variation in this systemic inflammatory response. And so moving forward, we're hoping to identify which factors are actually contributing to this response. Is it, for example, the degree of local inflammation? Is it disease severity at baseline? Or is it things like patient demographics? And of course, the end goal here is being able to identify patients who are at a high risk of having a systemic inflammatory response and modifying their treatment plan accordingly. For example, we don't want patients who are going to undergo a really large systemic inflammatory response to be undergoing full mouth debridement. But at the same time, we don't want to be avoiding full mouth debridement across every patient, especially those where it may be more convenient, if they're not at risk of having this really pronounced response. Of course, the other side of things, as we've seen in these recent slides, there's this weird sort of unusual reformation long-term in the subgingival plaque microbiome, where it appears to reform in some patients, but not others. And moving forward again, we're hoping to identify why this might be the case. And again, the sort of downstream end product of this is, do we need to be able to identify these patients at an early stage, if we can use the day 1 samples, for example, to try and predict which patients are going to have this disease-associated composition reform? And do we need to start introducing things like active compounds, probiotics, treatments that specifically target the microbiome as adjuncts into their non-surgical treatment plan? So there's a lot of work still to be done, which is hopefully taking us down some exciting avenues. So thank you very much for listening to me. Hopefully that was interesting or partially interesting. Thanks to Shauna, Alex and Gordon, who have all speaking today. And of course, thanks to everyone else who's helped me. Bob Rosier, shown in this lovely figure, works with Alex in Valencia, and we collaborate with them quite extensively, and all of the oral science research group members in our lab at Glasgow who work with me and Gordon. And of course, thanks to Dentsply Sirona, who funded this trial. So I've left my Twitter and email if anyone wants to get in touch. And of course, I'm more than happy to take any questions at the moment. So thank you.
Sorry. Thank you, Mr. William Johnston. Here are some questions from the audience. Do you consider GCF assay to be more accurate for assessing local inflammatory response than the salivary?
It's a really difficult one, because what you find with the gingival clavicular fluid is you can only sample a certain amount of sites, and these are quite obviously the deepest pockets in the mouth. So what you get a really good idea of is the sort of immune response within this very site-specific location. And this is what we found throughout both this study and previous studies, whereas the saliva is much more of a reflection of the total mouth. So for example, if you look at the level of different pro-inflammatory cytokines in the saliva and GCF, they actually don't correlate, because you're sort of sub-selecting the GCF for these deep sites. So I guess it depends on what question you want to be asking. If you want to be looking at associations with full mouth clinical parameters, then the saliva may be more reflective of this. But again, you've got the interference of different bacteria, for example, where if you want to avoid this, GCF would obviously be more relevant.
Okay. Anyway, you have also pointed out that immediately after the scaling, SRP, day 1, there is a spike in CRP in both hand and ultrasonic. So do you advise any anti-inflammatory for this first few days to prevent that, at least to take care of that spike?
Yes, it's interesting. So what we see is that some patients undergo this spike and some patients actually don't. So I think using a sort of blanket anti-inflammatory antibiotic adjunct would not be the right course, because this isn't happening across every single patient. I think what is really, really important, and hopefully what we'll be able to shed some light on, is being able to identify patients who are going to undergo this response. And in those patients, then we can start to think about different anti-inflammatory adjuncts, of course, that might be of interest to them.
Anyway, thank you, sir, for your interesting lecture, which forms a gateway to many research fields for periodontal therapy.
Thank you very much.
I now request Dr. Fayazuddin, Chairman Dental Scientific Committee, Silver Jubilee Celebration, Rajiv Gandhi University of Health Sciences, to hand over a certificate of appreciation to Mr. William Johnston.
Thank you, William Johnston, for your enlightening lecture. And it is my pleasure to hand over this certificate of appreciation for your contribution to the success of this symposium. This certificate is on behalf of Rajiv Gandhi University of Health Sciences. Will you please project the certificate? Let the audience have a look at it.
Thank you very much. And thank you for letting me talk today.
Yeah. So thank you very much. Yes. Well, thank you, Johnston, once again for your contribution to the success of this symposium.
It is my pleasure to introduce the last speaker of today's program, Professor Kalsha. She is a professor of periodontology and immunology at Glasgow Dental Hospital and School, Glasgow. She is an honorary consultant in periodontics, senior clinical lecturer, clinical research facility lead, and lead for undergraduate teaching in periodontics. During her undergraduate dental training in Glasgow, she completed an intercalated degree in immunology. These studies extended to an intercalated PhD examining the role of cytokines in inflammation. In 2011, she completed specialist training in periodontics and the following year commenced an honorary consultant post in periodontics, at the same time becoming the first dentist to be awarded the Scottish Funding Council's Senior Clinical Research Fellowship. Currently, Professor Kalsha treats patients referred for specialist periodontal care, leads a research group focused on local and systemic consequences of immune response in the oral cavity, and trains undergraduate and postgraduate students, NHS hospital and university staff. Today, she is presenting a lecture on local and systemic consequences of immune response in oral cavity. Over to you, ma'am.
Thank you very much for the very kind introduction. I don't know if you can see my screen. The screen I can see is the certificate, but maybe you can see my presentation.
Yeah, we can see your presentation.
You can see the presentation. That's wonderful.
Yeah.
Well, it really is an honor to have the opportunity to speak at this meeting today. Although I think I maybe have the most difficult task because I have to follow those last three speakers, which is going to be very challenging to do as good a job. So just by way of formality and for transparency, a brief disclosure of some of my funding sources, and then moving on really to, well, where I am is at the University of Glasgow. Our university is over 500 years old. We don't have the luxury of a lovely modern and new university. But nonetheless, like in all old universities, the clinicians and the scientists, they never work in the beautiful old buildings. They always work in the very modern buildings. And so this is where myself and Sridhar Rao, who very kindly extended this invitation, work here at Glasgow Dental Hospital. And I also do some work with colleagues in the Infection Immunity Research Institute. And when Sridhar first mentioned this to me, this was back in 2019. And I was very excited because I was really, really thrilled and privileged at the opportunity to visit the south of India, which is somewhere I've never been. And so this was a wonderful invitation to receive. However, as we know, times have changed a little bit. And so here I am in Glasgow now, and this is what we all look like. But I have to say, I think the opportunities and the innovation that has been sort of arisen from this situation has been fantastic and really exemplified by this absolutely brilliant meeting today, which has managed to bring together, I've thoroughly enjoyed the last three speakers, and hopefully the audience have too, and hopefully it's been a really interesting opportunity. So in spite of that, I think it's been a wonderful meeting and a wonderful day so far. So I don't think anybody in this audience probably needs much convincing that periodontitis, which is the disease that I am most interested in, is an important disease. Economically speaking, dental diseases are really important, and I think it's something which perhaps isn't recognized as much as it should be. And across most healthcare systems, spending on dental disease is around 5% of most healthcare budgets, and that's a lot. And worldwide, this is around $400 billion per year. Of those costs, you can attribute a substantial proportion to periodontitis and its sequelae, the consequences of tooth loss. And it's a really common disease. 50% of the population at least suffer from this disease, with 10% suffering the severe forms. And this arguably makes it the most prevalent chronic inflammatory disease, and arguably the most prevalent cause of bone loss in the human population, even above osteoporosis. And I think dental disease is sometimes perceived as quite far down the list of importance. And yet, when you look at some of the data, certainly the health spending in the EU, outside of cancer and oncology, ranked number 1 was cardiovascular disease, ranked number 2 was diabetes, and ranked number 3 for spending was dental disease. So economically, these are important diseases. But we also all know that on an individual level, having functioning teeth and gums is essential. It's essential for verbal and non-verbal communication, for socializing, for eating, for all these things that are absolutely essential to both physical and mental well-being. So for me, it's an important disease that it's well worth looking into. And this is what a lot of my patients look like. And I know that I believe there's quite a lot of periodontists in the audience and people who have an interest in this disease as well. And I'm sure your patients are similar. So this lady, she's only 24 years old. She has already lost 3 of her lower front teeth. She has gingival recession. Her front teeth have moved and drifted, causing problems with the appearance and arguably problems with eating. And yet this lady, her gingivae, her gums, they don't look massively inflamed. There is some inflammation at some sites that you can see here. And the other thing to note about this lady is that her teeth are not covered in dental plaque. There are not vast amounts of microbiome deposits here. And you can compare this to this person here, who clearly doesn't manage to brush their teeth as well. There are large plaque deposits throughout the mouth, and this is visibly having an impact. So there is florid gingival inflammation, there's red gingivae, inflamed gingivae, swollen gingivae in response to these plaque deposits. But this patient does not have periodontitis, they have gingivitis. their teeth are all still there. So even though they have this inflammation, they haven't had the tissue destruction that's caused the loss of tooth. And so we know that the microbiome is necessary, and the previous speakers have clearly demonstrated that the microbiome is complex, it has multifaceted roles. It's necessary, absolutely, but it's not sufficient for periodontal disease. So there's something else happening as well. And this is not a new idea. This study was almost coincided with the start of your society. But this study was one of the seminal studies that really suggested this from a long time ago. And what you see here is these three lines represent three different groups of patients in whom periodontal disease was rapidly progressing, moderately progressing, and non-progressing. And what this represents here is the extent of calculus and plaque deposits found around their teeth. And these are patients who were followed up over several years, with the researchers going back to visit the patients repeatedly over these time points. So they all kind of had fairly similar levels of plaque, but they certainly did not all progress at the same rate. What you found was that you have a subset of patients in whom disease hardly progresses at all. You have a group in whose disease progresses at a moderate rate, and then you have a third group in which the disease progresses rapidly. And over the last 3 decades, the field has really begun to drill down and understand why we see these three groups of patients and why perhaps the immune response is important in determining what happens when you encounter your microbiome in the oral cavity. So currently, we now have actually pretty good understanding of a lot of this. We know a lot about the microbiome in periodontal disease. We know a reasonable amount about the microbiome in gingival health. We know quite a lot about this immune response that you see within the gingival tissues, in the connective tissues. We know that there's a variety of immune cells that you find in there. You find huge numbers of different inflammatory mediators, that ultimately these activate osteoclasts, and the osteoclasts will cause bone destruction. We know that in health, you still find some of these mediators, you still find some of these cells, but they seem to be reasonably well controlled. They manage to strike a balance between what's happening in the tissues and what's happening in the microbiome, and the alveolar bone stays intact. And our mainstay of treatment for our periodontal disease is mechanically disrupting this biofilm. And that has reasonable results. It does work to a point. However, we all know and we all see patients in whom that doesn't really work that well. And so we start looking for different treatment modalities. We start looking for surgical options, for grafting, for dental implants, using local and systemic microbials. So our treatment options for periodontal disease developed over time, over and above that mechanical removal of the biofilm. And the reason they've developed is because that mechanical destruction of the biofilm isn't always sufficient to resolve the disease. So my hypothesis, if you like, is that if we can understand the immune response that's happening here, this opens up avenues for novel prevention and intervention for periodontal disease. So one of the first questions to ask is, well, what drives the immune response? And as we know, we've seen a lot about the microbiome and the different bacteria, and we know a lot about those bacteria, and we know a lot about what some of those bacteria can do to manipulate the immune response. And it's in their interest to manipulate the immune response, because for them to maintain their environment, they require inflammation. And they do this in different ways. So there is some element of specific immune response with specific antibodies, which Will showed earlier in his talk. But there are other things like complement manipulation, changes in TLR signaling. The AA can cause hyperactivation of neutrophils. It can generate altered proteins. There are a huge list of things that these bacteria can do, which have now been quite well characterized. And so particularly some of the bacteria that we know about here will themselves cause marked alterations in the immune response. So what can you do about that? Well, can you target the inflammation to prevent the tissue destruction? And if you're going to do that, there's so many things going on within this tissue and within this inflammation. How do you decide which one to target? And I kind of view this as inflammatory processes and this system, if you like, is a bit like an orchestra. So there are lots of different components and they all work together. And when they work together in such a way that they cause this alveolar bone loss, this activation of osteoclast, you get periodontal disease. So you have to try and persuade the orchestra to play a different tune. You have to try and manipulate it. You don't want to knock it out altogether because you need the orchestra to work, because otherwise your immune system's not going to work. So you have to pick which bit you're going to manipulate. And this is just an aside, but I had to share a flat when I was a student with clarinet players. So I would always target the clarinet part of the orchestra here. So one of the ways to decide which part to target is to use animal models. And Gordon mentioned earlier, this is something we've done a fair amount of. And this is the model that we use, or one of the models that we use. And this involves pre-treating mice with antibiotics, because the problem with mice is that they don't naturally harbor human periodontal pathogens. They do get periodontal disease as they get old, but that takes a couple of years. So if you want your experiment to be done a bit more quickly, you have to accelerate their periodontal disease. And one of the ways of doing this is infecting them by oral gavage with Porphyromonas gingivalis. And you need to give them the antibiotics because otherwise the Porphyromonas gingivalis can't find a niche within the oral flora. And what you see in these mice is that they lose alveolar bone, much the same way as humans do. And you can measure this. These are obviously rapidly, much enlarged from where they would be in a mouse. But you can identify a kind of amylocemential junction. You can identify the alveolar bone, and you can use software to calculate how much bone loss you've got. And you can also do kind of cone beam CT, micro CT imaging to establish how much bone's been lost. So that's the model that we use. And by using mice, there are all kinds of tools available that allow you to study particular components of the immune response. And one of the ones that one of my colleagues, Jennifer Malcolm, has looked at is the effect of mast cells. And this was quite surprising to me because I wouldn't assume that mast cells would play much of a role in periodontal disease, but we actually found cytokines derived from mast cells and also activated mast cells in gingival tissues. So we wanted to ask what role they were playing there, because mast cells are assumed to play a role in being sort of sentinels of mucosal surfaces and preventing infection, if you like. So we thought, well, what are they actually doing? So what we used to investigate that was a type of mouse, which is called a KIT-W mouse, which has a naturally occurring mutation which prevents the mouse from developing mast cells. So the mouse is in effect devoid of mast cells. So that's what these mice here are. And we have our sham control mice, which means they just get the carrier, they don't get any Porphyromonas gingivalis. We have our normal mice that are infected with Porphyromonas gingivalis. And on the y-axis, we have the bone level. So the closer to the x-axis you get, these mice have bone loss, basically, compared to their controls. But what you see is when you try and infect the KIT-W mice with Porphyromonas gingivalis, they don't get any bone loss. So rather than being a defense mechanism and preventing the Porphyromonas gingivalis from taking hold, it seems that the mast cells actually prevent the inflammation. They prevent the inflammation and the destruction. So this is kind of interesting. And we went on to look at a few of the cytokines. So this was a cell that we had removed from the mouse. And we went on to do a series of experiments looking at some of the cytokines derived from mast cells and that are involved in mast cell activation. And one of those is a cytokine called interleukin-33, which acts as an alarmin. And it was particularly interesting to us because it had shown to have, in some situations, it protected against osteoclast activation. And in other situations, it will actually promote osteoclast activation. So we thought, well, this is kind of interesting, and say it's been known to activate mast cells as well. So we were interested in that. So what we did here was we administered this cytokine to the mice. And I haven't shown the data on the graph because it gets very convoluted, but if you give the cytokine as well as the bacteria, you get a little bit more bone loss. If you then give another cytokine, which is called osteoprotegrin, then this will block that effect. So the mice given this cytokine, which prevents osteoclast activation by blocking the effects of rank ligand, if you give them this cytokine, it doesn't matter what you do to them, you can give them other cytokines, you can give them Porphyromonas gingivalis, then you don't get bone loss. So the immune system at that site, at the oral mucosal site, can be manipulated to prevent the bone loss that you see when you infect mice with Porphyromonas gingivalis. And these are just some examples. There are dozens now of examples of different studies looking at this. Some of them have gone to some clinical trials, and I would recommend this little review article here, which really highlights some of the ones which are perhaps coming to the forefront. The sort of pro-resolving mediators have been around for a long time, but there are now lots of different pathways that we know enough about that we can target them. So this is something which I think is not quite in routine clinical practice yet, but it's something that as we understand more and as we learn more, we can begin to get a handle of. So that's just a little bit about the sort of local destruction and the local inflammation. And for me, it's clear that this is an avenue that we really have to explore outside of our current avenues of implants and bone grafts and antibiotics. So the other thing is that we have to consider is that if you have inflammation at a site, and if you have periodontal disease, that site can be quite large. So within a patient with periodontitis, you can estimate that this site of ulcerated epithelium here, where you know there's this active and ongoing inflammatory response, that site can be about 20 square centimeters, which is quite large. It's about the size of the palm of your hand. So if you have inflammation on that scale, on that level, it doesn't seem a big stretch to assume that the impact of that inflammation might not be limited to the mouth. And this is where the idea of the systemic consequences of the immune responses in the oral cavity comes in. And this is something that we've been interested in, particularly in the context of rheumatoid arthritis and how autoimmune reactions might be triggered and perpetuated at mucosal surfaces. But I'm going to try and give a sort of brief overview as to what I think is happening at the moment and where this field is at. So again, this is something which has grown and grown and grown over the last 2 or 3 decades. And this was taken from a recent review from George Hadjish and Gallus. And this has drilled down into the periodontal comorbidities. So this is the list of diseases which is growing and growing, which are associated with periodontitis. And in this list, the list has been a bit restricted to the ones which have more reasonable epidemiological evidence behind them. So we have things like Alzheimer's disease, aspiration pneumonia, non-alcoholic fatty liver disease, inflammatory bowel disease, rheumatoid arthritis, colorectal cancer, diabetes, cardiovascular disease, and adverse pregnancy outcomes. So it's a fairly long list already, but it is still growing. And the next thing that they've highlighted here is that in some of these diseases, in some of these disease states, it's been reasonably consistently demonstrated that you can find either bacteria that originate in the mouth or their DNA in the site of disease. So as an example, you will find oral bacteria in aspiration pneumonias. We know that things like Fusobacterium nucleatum may translocate to the gut and therefore may have this role in colorectal cancer. We know that this bacteremia that Will has mentioned and that others have mentioned, we know that that seems to be something which might contribute to the adverse pregnancy outcomes, to the cardiovascular disease. It might contribute to Alzheimer's disease, as studies have shown that you can get fragments and components of Porphyromonas gingivalis that are able to cross the blood-brain barrier. So there is this microbial translocation component. There's also this notion of a host response component. So there's two things going on here. There's bacteria that leave the mouth, But there's also the host response in the mouth that isn't just restricted to the mouth. So you have a common mucosal immune system. So the immune response in the mouth isn't just going to affect the mouth, it's going to affect the rest of the mucosal immune system. And there was a really elegant paper in Cell recently which showed that if you activate T cells in the mouth, those same T cells, those exact same T cells, can contribute to damage in the gut. That's in mouse models, but nonetheless, it really highlights the potential impact of this. And so the host response elements that link these diseases, we know we see endothelial dysfunction, which is rightly relevant to cardiovascular and other diseases. We know that we see metabolic alterations. Plenty of people have shown that patients with periodontitis may have an elevated HbA1c. We know that we get class precursor priming and altered hematopoiesis in the bone marrow in patients with periodontal disease. So you see a slight change in the stem cell compartment. And then the idea of this chronic low-grade inflammation. So in some studies, patients with periodontitis have slightly elevated CRP, slightly elevated serum cytokines. And that of itself will contribute to these diseases such as cardiovascular disease and endothelial. So it's a complex story, but what's really exciting at this point in time is that we have the technique tools to start to unravel it. And by unraveling it, we can understand it. And in doing so, we actually potentially, even by working in the mouth, have the number of different oral diseases. So I'm just going to talk a little about the epidemiology. And this has been hugely studied. And I'm going to start with a study, just because it's local to us, a heavily cited study. And I'm starting by talking about cardiovascular disease, because that's probably the comorbidity that has received attention over the years and has been surrounded in lots of controversies along the way. And so this study, 12,000 Scottish patients, and it's quite a good place to do a study because our healthcare system allows us to track patient data very well. We have a patient population who don't necessarily have the healthiest of lifestyles. So your disease incident rate is reasonably high. But what they found in this population is that patients who brush their teeth once a day or less have an adjusted hazard ratio for cardiovascular disease of 1.3. So by brushing your teeth less than once a day or once a day, you're 30% more likely to have a cardiovascular event. Now, it doesn't prove causality, of course, but it does start to suggest that oral health is not an isolated thing. This study was a little bit different. So this one took an approach of looking at patients who turned up at the dental hospital in the Netherlands, in Acta, and they managed to find 60,000 patient records. And within those patient records, because they were a dental hospital, they could quite accurately identify which patients had periodontitis and which ones didn't. So of their patients who had periodontitis, nearly 5% reported cardiovascular events. Of all their other patients who were coming into the hospital, it was less than 2%. So they were about, the periodontitis patient population was about 60% more likely to have a cardiovascular event in this study. And Thomas Dietrich took a systematic review of this a few years ago. And basically, it's not 100% consistent across every study, but 11 out of 12 studies reported positive associations between periodontal disease and the incidence of acute cardiovascular disease. And the association is slightly stronger in younger adults. So I mentioned that our interest particularly is in rheumatoid arthritis. And so the comorbidity here is that patients with periodontitis might be at slightly elevated risk of getting rheumatoid arthritis. I'll come on to that in a little minute. But the other thing to note is that there is a dose response in all these relationships. So within the cardiovascular field, when you start to do sort of subgroup analysis, you find that the patients with severe periodontal disease have greater risks. And you clearly see that, I think, in this study with patients with rheumatoid arthritis. So the worse the periodontal disease, the greater the alveolar bone loss. This tender joint count, the hospital-acquired disability score, And this measure of autoimmunity, this anti-CCP antibodies, all of these parameters were substantially worse in the patients who had concurrent periodontal disease. So the more periodontal disease you have, the more rheumatoid arthritis you have. The more rheumatoid arthritis you have, the more periodontal disease. So taking a slightly broader view and looking at a select retrospective based studies with slightly different designs, but nonetheless, and these are, I think, almost all across the Taiwanese population that have a very nice record that allows them to do these studies. And over the last few years, they've really pulled the periodontal disease into these studies. And what you see is it's quite consistent. The sort of increased risk of morbidity of all sorts of different diseases is consistently a little bit more if you have periodontal disease. So your major adverse cardiovascular events, a little bit more. Your rheumatoid arthritis, almost twice as likely. Your Alzheimer's, a little bit higher. The bipolar disorder, which in this study, actually, they also reported diabetes, ischemic heart disease, stroke, chronic obstructive pulmonary disease, and asthma. And even peptic ulcer disease. Now, this is a very small increase, but nonetheless, it's an increase there. And the same for glaucoma. So it almost begins to look a bit of a kind of generic thing. If you have periodontal disease, you're more likely to be unwell in many, many different ways. So we see all these associations, and I think we have to be a little bit cautious with our interpretation of them, and particularly in how we're going to move forward as a field, knowing all these associations. And what this shows is two penguins who are having a little chat, and they're asking themselves, do you think all these film crews arrived because of global warming, or did global warming make all the film crews arrive? And we have this same problem with all these association studies. We need to understand where the different diseases fit within this model of all these different inflammatory diseases. So right now, I think we're almost as confused as these penguins are. But hopefully, as time goes on, things are going to become a bit clearer. And I would highly recommend this little opinion piece, which is actually very recently published in the Journal of Dental Research. And it really highlights that, and it's not to criticize people's studies at all, it just highlights how difficult it is to do these association studies and to do these population-based studies. It is incredibly difficult because human beings that live in society are complex. They're not mice in a cage. You can't control for everything. And so we have to take that into account when we're interpreting the data. And this article puts, I think, that into quite good terms, almost certainly better than I can. So we see all these associations. We see that, you know, if you've got periodontitis, there's a good chance that you'll pop up somewhere else in this diagram here as well with one of these other conditions. And there's a couple of important questions, I think, that should be answered, not just for us as people that are interested in the mouth, but for the world of healthcare in general. The first one is, how causal is periodontitis in any of these comorbidities? And that's difficult to ask. And then linked to that really is, well, if it, you know, what is the mechanism that links them, and is it a causal mechanism? So this is where it gets quite complicated. And I think the complexity is quite well highlighted in this diagram here. This was taken from a recent review about autoimmune mechanisms only. So this is just one mechanism. And so you see all kinds of different things, and you can find evidence for molecular mimicry. You can find evidence for superantigen activation of T cells. You can find evidence for autoantigen production. You get autoantigens produced in the gingival tissues. You can find microbial translocation. You will find bits of Porphyromonas gingivalis in a rheumatoid arthritis joint. We know that circulating myeloid dendritic cells have live P. gingivalis in them, and we know that those dendritic cells go to different places. We know that we have excess production of cytokines, particularly in some patients. So it's quite complex. And I think that makes it very difficult. And some of the evidence for this is drawn from mouse studies, and some of it's drawn from human studies. And so it's very difficult to piece this all together. But we're beginning to get a map, and albeit a complicated map, I think we are beginning to get a map. And as I say, it's complex and it's not perfect, but we are beginning to see a map. So the way I view this, which is perhaps a bit of an oversimplification, but the way I view this is that we know that locally we have this microbiome, which is dysbiotic. We know that we have excess cytokine production. We know that from this dysbiotic microbiome, we have that microbiome associated with ulcerated epithelium. And so we absolutely know that we get a bacteremia in patients with periodontitis and in patients who don't have periodontitis, but more so in patients with periodontitis. So we know we have this circulating bacteremia. We also know that we have these cytokines, and we know that there are elevated levels of those cytokines within patients with periodontitis. Not all of them, but some of them have elevated systemic cytokines as well. So we know that that's there at a local level. What we also know, and what we have seen fairly consistently, and it's kind of assumed that, maybe this is downstream or maybe it's related, we know that you can swallow these bacteria. As Alex Mira mentioned, you swallow a liter of saliva a day, and in amongst that, there's a whole lot of bacteria. And what has been increasingly seen, again, predominantly in mouse models, but it has been seen, is that you get gut dysbiosis. So you don't just get dysbiosis of the oral microbiome in periodontal disease, you get gut dysbiosis as well. We and others have seen that you get protein modifications in the gingivae. You get autoantibodies. You get antibodies against various cellular components. You also get antibodies against modified proteins, such as citrullonated proteins. And these antibodies are associated with rheumatoid arthritis. And people like Joe Conkle and others have shown that you get this kind of altered stem cell compartment. So there is something happening in the bone marrow, which is altered in periodontal disease, because the stem cells change as well. So all these things are happening. Ultimately, any and all of these things can lead to this aberrant immune system activation, which is a very vague and generic term, but one way or another, your immune system has got activated in such a way that it's not helping you out that much anymore. So we know that the bacteremia can do this, the gut biosis can do this, we know that modified proteins can do this, we know that autopod hematopoiesis can do this, and consistently chronically low-grade elevated cytokines will contribute to all these things. So they're all a little bit intertwined. And so they have this kind of network of events going on, which, as I say, is complex and difficult to understand, but we do have now the tools to start to pull it apart. This idea of aberrant immune system activation is quite hard to measure. It's very hard to put one measure that says your immune system isn't quite doing what it should be doing. There's not really a single molecule or a single cell or a single thing that you can measure that says you are suffering in some way from this aberrant immune system activation, much less to say which part of this is actually causing it. So what the field has done, not just in periodontal disease, but in other diseases as well, is to take C-reactive protein and use it as a surrogate measure. And CRP is a far from perfect surrogate measure, but it is useful because it's got a relatively short half-life, and it's produced in response to inflammation, to some sort of insult that stimulates the liver to produce CRP. And if that insult is removed, then you stop generating excess CRP. So if you were to go and run a marathon, you would find that your CRP will be elevated a couple of days later. But assuming you don't continue to run marathons every single day, or as you do so, your body adapts to it, that CRP will stop being elevated. So it's something that changes, and that's quite important if you want to measure these things. Huge, enormous cardiovascular studies have come up with this idea that you can estimate your cardiovascular disease risk. And again, it's not perfect, but it's an estimation based on your circulating CRP, your serum CRP. And that can be low, medium, or high based on how much CRP you've got. So where does periodontitis sit within this? Well, if you look at periodontitis, it very much depends on the periodontitis that you measure. So for some patients, the periodontitis, the CRP is generally less than 1, so it's pretty low. In other studies, the CRP, I think the highest I could find reported was about 7.9 as a baseline kind of resting CRP in patients with periodontitis. This isn't after treatment. This is just a patient with periodontitis. So that's really high. If you look in patients with diabetes, it's a little bit more consistently elevated, but again, the range is really high, often depending on obesity and blood sugar control. If you look at patients with obesity, again, some of them have pretty normal levels of CRP and some of them it's quite high. Even osteoarthritis, which you wouldn't necessarily consider to be an inflammatory disease, it's a disease which is generally, kind of written off as being wear and tear, but there is an inflammatory component to it. Patients with osteoarthritis will have an elevated serum CRP, as often do patients with rheumatoid arthritis. So CRP has its uses. And Will's already mentioned that we've used that to estimate the kind of inflammatory insult that you get when you treat patients and how you get that spike. But what we're looking at here is what you get before you treat them. So, as mentioned, that range in the patients with periodontitis is huge. So, for patients with a low PISA, and this is a periodontal inflamed surface area, so this is a composite measure looking at the probing pocket depth and the proportion of sites that bleed on probing. So if you look at the patients with the most severe disease, you find that their CRP is elevated. And when you look at this in relation to the sort of thresholds, you find that, so this threshold of 1 is considered that low cardiovascular disease risk. The 1 to 3 is considered moderate, and the over 3 is considered high. So this was taken from Will's study, which I have to emphasize was a population of otherwise healthy people who had no medication. They had no known medical conditions. They were fit and well. We weren't able to extensively screen them. We had to rely on self-reported screening, but they were essentially reported as fit and well. And I think the average age was in the early 40s for this study. Will can correct me if I'm wrong there. But even so, even within that group, there's a subset of those patients that have got a CRP which is which is putting them into a risk group for cardiovascular disease over and above what you would see in healthy people. And this systemic inflammation, this CRP, this notion of the systemic impact of periodontitis, this was taken from the Journal of Clinical Pathology summary of the classification of periodontal disease from 2007. And I'm just going to zoom in on this bit here. So this isn't routinely used clinically. I don't know of any periodontists who are currently measuring CRP in their patients. But I think the point is that it's a concept and an idea and something that we've found which is felt to be of sufficient importance that it has been added in to the classification system as a means of future-proofing that system. So what they have estimated then is that, your sort of grade, if you like, your severity, your impact of, how you can modify your periodontal disease, how serious is your periodontal disease. You could take a measure of CRP to feed that into it. So there are other things here, like if you have diabetes, how poorly controlled is your diabetes? But even for patients without diabetes, what if their serum CRP is elevated? What if they're in this greater than 3 milligrams per litre, which a small number of our patients were? What if those patients, they're suddenly in a high risk for cardiovascular disease, in some cases without any known comorbidities? Some patients will have comorbidities, but not all of them. Should we treat them differently? Should we do something differently? How do we manage that? Can we follow those patients up differently? So for me, I think the fact that this idea of the systemic inflammation, or our proxy for systemic inflammation, has made it into the classification system suggests that this is something that we really are going to have to consider. We can't kind of just continue to ignore that periodontal disease is not just limited to the mouth. And just to kind of wrap up a little bit, idea of CRP being important and it being worth considering when you're looking at your disease and looking at your disease severity is not new at all. So this is a study published more than 10 years ago. And what this study looked at was 15,000 people who were basically healthy, but they had elevated serum lipids. So they had a lipid profile which put them at elevated risk of cardiovascular disease. They haven't had any cardiovascular disease yet, but they looked for increased serum lipids, but they also looked at their serum CRP, okay? So it could be the serum lipids or elevated CRP. And they took these 15,000 people and they gave them a high-dose statin. And statins, as well as modulating lipids, we now know increasingly have quite an impact on inflammation. And what they found with this was that you get up to a 60% reduction in vascular events compared with placebo 4 years later. So that was quite a striking finding. And this was one of the studies that was using CRP as a therapeutic target. And more recently, this was kind of substantiated as an approach. So this was a trial called the CANTOS study. And this one used a monoclonal antibody to neutralize interleukin-1. And this study was a slightly different population group. So this had 10,000 patients with a previous myocardial infarction whose high sensitivity CRP was greater than or equal to 2 milligrams per liter. So clearly their risk profile is a bit different because they've already had one significant cardiovascular event. But all they've done here is target inflammation. There's no direct targeting of lipids, which is the kind of conventional approach. There's no direct targeting of other risk factors. All they have done is go after IL-1, which is probably one of the sort of seminal inflammatory mediators. And certainly in our hands, we find that IL-1 really consistently associates with disease severity, and it's consistently modulated by periodontal treatment. So if your treatment's successful, your IL-1 reduces. So after 4 years, they found between a 20 and 40% reduced incidence of cardiovascular events, which was felt to be quite a significant reduction. Now, the problem with giving people these kind of therapies is that firstly, they're very expensive, but secondly, they do carry side effects, and particularly at higher doses, they carry side effects. So using monoclonal antibodies to block your inflammation, it works, but it's maybe not ideal. But maybe if somebody had checked these patients for periodontal disease, maybe they could have brought their CRP down by reducing that as well. So just by way of conclusion, I think it's really worth noting that the inflammatory burden of periodontitis now features in the disease classification system. I don't think it's going to become routine clinical practice soon, but I think it's something that we should consider. I think the systemic impact of periodontitis is complex, clearly, and we don't fully understand it yet, and we don't fully understand the mechanisms yet, but there is huge progress being made. There is this systemic immune system impact, and we see that in our studies, plenty of other people see it in theirs. And it's that systemic immune system impact alongside the bacterial effects that might account for these associations between periodontitis and other diseases. Looking locally in the mouth, for me, and this is my feeling and my opinion here, is for me, for sustainable treatment for periodontal disease and patients with periodontal disease, antibiotics, surgery, bone grafts, and implants, they are treatment options that we have available to us now. But this is not the optimum solution. As Alex already mentioned, dentists, well, the antibiotic resistance problem is enormous and growing. And dentists currently prescribe across most healthcare systems about 13% of all the antibiotics that are prescribed across the whole of medicine. That is a lot. So for us to say that this is a mainstay of treatment and what we plan to do for the future, I don't think is a good idea. Carrying out bone grafts and implants in patients can be hugely effective. But the issue you have is that if you can't control this local immune response, if you can't rein in the thing that caused the problem in the first place, then your implants are going to get exactly the same problem. And we see this epidemiologically. We see that patients with periodontitis are more susceptible to periimplantitis and implant loss. Our animal models and some early human studies are beginning to show us that targeting that local immune response can control the local tissue destruction. And doing so, we just might be able to control the systemic manifestations. So, oh, my apologies. That's just a brief notion going back to this idea of antibiotic surgery and bone grafts, that the treatment stage guidelines that the European Federation have produced recently in response to the sort of new classification system, it highlights the need to control risk factors as the first stage. So things like poorly controlled diabetes. Now, at this point in time, we don't have a means to directly control the local inflammation. But I think as we're changing our approach to treatment, we're beginning to highlight and understand that it's not just about removing the biofilm. There are other things that we have to play into too. So again, this is a really interesting document, and looking at the evidence, they've really trolled through that behind, why it's so important that we control the local factors and the systemic factors in our patients before we even attempt treatment. So it just remains me to thank all my friends, my colleagues, my collaborators. The work I showed was done by a number of these people. Jennifer Malcolm did a lot of the animal studies, but the rest of the group over time, some of these are previous and past members. I have great pleasure to be able to collaborate with two brilliant microbiologists, which has been such an education for me over these years. And obviously I need to thank the people who have paid for the work as well, and to say thank you very much for your attention, and I would be happy to take any questions.
Thank you, Professor Kalsha. Here are some questions from the audience. What is the contribution of immune response versus the microbiome in development and progression of periodontal disease? How much to blame the immune response?
So I guess that is the sort of age-old question, and I think it depends who you ask. So it's very difficult to get bone loss in mice that have compromised immune systems, almost no matter what bacteria you give them. And I think it is very much for want of a better analogy, a chicken and egg analogy. So the microbiome that we study, that Will has shown, that Alex has worked with us to really very nicely characterize and show how it changes, that microbiome only exists in a particular niche, and that particular niche requires inflammation. So yes, I think that's a very difficult question. I think we're beginning to understand it, and I think we're beginning to see how different individual bacteria might play a role. But then when you extrapolate that to patients, it becomes very difficult, because we know that the microbiome in patients is a bit varied. Not all patients with periodontitis have P. gingivalis, and even some people with gingival health have P. gingivalis. So it's a difficult balance. I would be happy to invite any of the previous speakers to answer that question as well.
So have we reached a stage to personalize periodontal therapy?
I don't think so, not yet. I think we have to keep a close eye on our colleagues who work in fields like rheumatoid arthritis, in inflammatory bowel diseases. I think they are getting closer to that. And I think there are enormous lessons that we can learn from those fields. And I think perhaps we might get there at some point. I hope it's going to be soon, because I'd really like to see it. But I think we want to keep a close eye on some other fields, because if you think about what the gastroenterologists do, you don't see people with Crohn's disease and colitis. They don't really worry about targeting their microbiome. They target the immune response, even though those patients have a terribly dysbiotic microbiome. The rheumatologists have managed to stratify their disease into the anti-citrullinated protein positive and ACPA negative patients, and those patients are now being treated slightly differently, as I understand it. So I think there's a lot we can learn by looking to other fields.
So cytokine-related therapy is has more promise in the control of periodontal disease management?
So in terms of things like the monoclonal antibodies that the rheumatologists use and the cancer field uses and things, I think there's work to be done there. I think they've shown pretty tremendous results in other diseases. when they're targeted to the right patients. And I think we can learn from that.
Thank you, Professor Kalsha, for a better understanding of etiology and pathogenesis of oral diseases, which is an ongoing process. I now request Dr. Fayazuddin, Chairman Dental Scientific Committee, Silver Jubilee Celebration, Rajiv Gandhi University of Health Sciences, to hand over a certificate of appreciation to Professor Kalsha.
It was a very thought-provoking lecture, Professor Kalsha. You have touched upon the very final aspect of immune responses, impact of immune responses on the systemic impact. And in appreciation of your contribution to the success of this program, it is my pleasure to hand over you this certificate on behalf of Rajiv Gandhi University of Health Sciences, Bangalore Academy of Periodontology, and VS Dental College. Thanks a lot, madam. We are very grateful to you. Thank you.
Thank you so much. It has been a real privilege and an honor to speak to you today. And I hope we can continue to keep the conversation going, because we have all the technology now, and it's been an absolutely wonderful opportunity. So thank you.
We should continue this in the future also. Thank you.
I now call upon Dr. Rekha Jagdish to deliver vote of thanks.
They say gratitude is the healthiest of all emotions. I'm here today to express my heartfelt gratitude to all the people who are involved in executing this virtual symposium. We had around 760 registrations. It is my privilege to thank Rajiv Gandhi University of Health Sciences, Dr. S. Sachidanand, Honorable Vice Chancellor, RGUHS, Sri Shivanand Kapashi, Registrar, Dr. N. Ramakrishna Reddy, Registrar for Evaluation, Mr. Manjunath Hegde, Finance Officer, and Dr. Mohammad Fayazuddin, Chairman, Dental Scientific Committee, Silver Jubilee, for supporting this prestigious event. I would like to thank Bangalore Academy of Periodontology advisors, Dr. T.R. Gururaja Rao, Dr. C.D. Dwaraknath, and Bangalore Academy of Periodontology coordinators, Dr. A.V. Ramesh and Dr. M.V. Ramachandra Prasad, for organizing this program. I'm thankful to the management of Vokkaligara Sangha Dental College and Hospital and our principal, Dr. Rajkumar GC, for fostering the sense of support and encouragement. The valuable contributions of Dr. Meera, Professor Ramesh, Professor Kalsha, and Mr. William Johnston is really appreciable. We are truly grateful to you all, sir. I take this opportunity to thank all the participants for their patience, listening, and active participation. This event would not have been successful without the tireless efforts of the moderators of this event, Dr. Sushma R. Galkali, Dr. Aruna DR, and Dr. Sridhar N. Rao. I'm thankful to all my colleagues, Dr. Vinayak, Dr. Avinash, Dr. Sandeep, and Dr. Soumya, for their support and contributions for this event. Let me express my special thanks to Dr. Rajiv, the Dynamic Secretary of Symposium, for his hard work and commitment to make this program successful. Last but not the least, I thank Dr. Reena Kumar and the team to accord this IGCF platform for this enlightening event. Thank you, IGCF, for being with us throughout this event. I consider myself privileged to make a difference today and express my gratitude through vote of thanks. Participants can download their certificates from the link provided in the chat box. Thank you all. Good day.
Thank you, one and all. Thank you, Dr. Reena. Shreyansh?
Dr. Sushma, can you announce the next BAP programs?
Yes, sir. So it's going to be on 9th, right? 9th. Is it on 9th?
9th of April.
9th of April.
Yeah, 9th of April. as well as Perio Panorama 16th, 17th, 18th.
We are going to have some more events coming up. Next week, we are having a Perio Panorama, which is a rapid review program for the postgraduate students in the periodontology. It is mostly for the batch which is appearing for the exam, but anyone is free to register for the program. The program is going to be held between 17th, 18th, and 19th of March. And this will be followed by another informative session on, I think it is on clinical audit. It is going to be on April 9th.
Quality improvement and clinical auditing.
Quality improvement and clinical audit. The details of the program will be announced shortly. Thank you, Dr. Reena. It was wonderful to have support throughout. I really appreciate your efforts and your team efforts. Thank you so much.
Thank you very much. I was on the participant side all the time listening, so I joined in here. Shreyans has been here, and I'm so pleased that it went off very well, and it was an amazing, session. And I think this is so well positioned with wonderful talks. I've been lucky to listen to all the speakers, and thank you for making me a part of it by default. Thank you so much.
And about 760 registration. This is for the information of the resource person. At any given point of time, we had more than 400 people logged into these lectures. Thank you for the wonderful program.
Can I come in?
Yes, please.
I take this opportunity to thank each and everyone, those who have helped this directly or indirectly to make this program a success. Thank you very much.
Sir, can I put a word?
Yeah, sure.
We immensely thank our own Sridhar Rao.
Yeah, we have to be very grateful to him.
Who brought this program together. Thank you, Dr. Sridhar.
Sridhar, are you there? Thanks a lot. Thank you very much. God bless you.
Thank you all, sir. Bye, sir.
Bye.
Can we go offline, Reena?
Yes, yes. End the meeting.
End. Okay.
Yeah,
yeah. Thank you.
Thank you.
Thank you, sir.
Tthank you, Dr. Shona Kalsha, for Professor Kalsha.
Thank you.
Thank you, everybody. I know how much work it is to organize these, so thank you so much. Really appreciate the invitation.
Okay, thank you.
Thanks, Gordon. Thank you very much.
Jason.
Alex.
Anyone buy
Should I end?